So as soon as we go live, I'm gonna give you guys a heads up, and I'm gonna just share the link if you guys want to redirect it. So, okay, so we are live. So be all welcome to our first thematic symposium in the Science Clubs Brazil. So Science Clubs Brazil is part of the Science Clubs International. That's a big hub for scientific outreach, as well as teaching students from high school and early undergrad studies how to do science in the best fashioned way. So today we have 4 special guests for our meeting. 2 from Brazil, 1 from the United States, and 1 from Canada. We like to introduce our first guest. She's from Brazil, Dr. Fabiola Ribeiro. She has a bachelor's degree in pharmacy by the Federal University of Ouro Preto and a PhD degree in biochemistry and immunology by the Federal University of Minas Gerais. She's currently an associate professor at the Department of Biochemistry and Immunology of Brazil, of UFMG in Brazil, and her research focuses on investigating the cellular and molecular neurobiochemical mechanisms inherent to Huntington's and Alzheimer's disease, and also schizophrenia, multiple sclerosis, and Zika virus infection in the brain. Dr. Fabiola will give us a speech entitled mGluR 5 Role in Neurodegenerative Diseases. So welcome, Dr. Fabiola. We are happy to have you here. You can share your screen, and we are going to start our first presentation right now, whenever you are ready.
Thank you, Pablo. And thank you for organizing this, and all the others, Bruna, and I'll start giving my talk now. Can you see my screen? Okay.
Yes, you can go on.
Okay, so today I'll talk about mGluR 5 and Huntington's disease, and I'll talk more about cell signaling implications of this receptor in Huntington's disease, or HD. So Huntington's disease is a neurodegenerative disease, and its main symptoms involve motor disturbance. And we can see here in this video, this lady has the involuntary and irregular movements of arms, legs, and neck. So this is quite common in HD. It's also called chorea dance, or choreic movements. And patients also have problems walking, eating, and speaking. And Huntington's is also a type of dementia. So patients exhibit cognitive decline as well as psychiatric alterations, as depression and psychosis are more common among HD patients than others. So what causes all these symptoms? So Huntington's disease is characterized by a specific neuronal cell loss. So here we can see a brain, or a post-mortem brain of HD patient. In this region that's called caudated putamen in primates, or striatum in rodents, we can observe that these regions is pretty much all one. This caudated putamen is mostly constituted of medium-sized spinal neurons, which are GABAergic neurons, and these neurons die specifically in the disease. And the cause of this neurodegeneration is a mutation in the gene that encodes the huntingtin protein. So when the huntingtin protein has more than 36 glutamines in the amino-terminal region, it will lead to the disease. So the data I'll show you today will be about characterization of the mGluR 5 dependent cell signaling pathways that are involved in Huntington's disease. So why to study mGluR 5, or the metabotropic glutamate receptor 5? Well, in Huntington's disease, well, this receptor is expressed in the main regions in the brain affected by HD, including striatum, cortex, hippocampus, hypothalamus. And here we can see these primary neuronal cultures obtained from mouse embryos that mGluR5 labeled in red and huntingtin labeled in green are present in the same neurons, the medium-sized spinal neurons, and they actually co-localize. Also, we have shown before that by co-immunoprecipitation experiments that the receptor can physically interact with huntingtin, and it interacts with both wild type and mutant huntingtin. And to study Huntington's disease, we have different mouse models in the lab. One is the BACHD mouse, which is a transgenic mouse model. So BACHD expresses an extra copy of the huntingtin gene contained, and it leads to the expression of a huntingtin protein containing 97 glutamines in the aminoterminal region. And we also have in the lab the Q175 mice, which is a knocking mice. So the first exon was replaced by a human exon, and the huntingtin protein in this case has 175 glutamines in the aminoterminal region. And we have also crossed these BACHD mice with mGluR5 knockout mice to further explore the role of the receptor in the disease. So mGluR5 has a quite complex cell signaling, and we can notice that as a G protein couple receptor, activation of mGluR5 leads to the uncoupling of the GQ proteins and activation of phospholipase C, which breaks down PIP2 in the membrane, forming IP3, which can bind to its intracellular receptors and lead to the release of calcium from these intracellular stores. And we and others have shown that in EHD, there is an increase in calcium load in the neurons due to the sensitization of many different receptors. So this increase in calcium could could contribute to excitotoxicity. But we also know that calcium is quite important for neuronal physiology. For instance, calcium can activate kinases such as PKC, which can activate MEK, ERK, and ERK is quite important for activating the expression of genes and fostering cell survival. mGluR5 can also bind to other proteins, including HOMA, and by binding to HOMA, it can activate ERK as well as PI3 kinase and AKT. And AKT is also a kinase quite important in EHD, and also because it can lead to neuronal survival. But it has been shown that mGluR5 can also activate toxic pathways. For example, mGluR5 can couple or can interact with PRPC, the cellular protein, that when bound to A-beta oligomers can lead to the activation of FIN, which is a SERC kinase, that can lead to an increase in intracellular calcium and activation of transcription factors that leads to translation dysregulation and can contribute for synaptic loss and neuronal dysfunction. So it really depends which cell signaling pathway is being activated by the receptor to see what would be the outcome. In terms of cell signaling alterations in Huntington's disease, it is well known that huntingtin can alter CREB and REST. So talking a little bit about CREB. So CREB is an important transcription factor that has to be phosphorylated and bound to CBP, the CREB binding protein, in order to increase the expression of its target genes. And these target genes are quite important for neuronal survival. And I have two examples, BDNF and ARC. However, in HD, mutant huntingtin traps CBP in the cytosol, and CBP can no longer translocate to the nucleus, so we have a decrease in the expression of CREB target genes. And also REST, which is a repressor of the expression of many genes. Wild-type huntingtin can bind to REST and can trap this repressor of transcription in the cytosol. However, mutant huntingtin is unable to trap REST in the cytosol, so now REST can move to the nucleus and repress many genes, some of them also important for neuronal survival. So as mGluR5 can modulate many different cell signaling pathways, we decided to investigate whether the receptor could modulate REST expression. And to test that, what we have done is employed primary neuronal cultures from mouse embryos, and we have stimulated these neurons with DHPG, which is a mGluR5 agonist, or blocked mGluR5 with CTEP. And what we can observe by Western blot is that REST expression is increased by DHPG. And one of its target genes is SNAP-25, the expression is decreased. And when we block the receptor with CTEP, we see a decrease in REST expression and an increase in SNAP-25 expression. So this data suggests that mGluR5 can modulate REST in primary neuronal cultures. But how could the receptor modulate REST expression? Well, it has been shown that REST expression can be modulated by the pathway involving N-cadherin and beta-catenin. So normally, beta-catenin is associated with N-cadherin, but if N-cadherin is phosphorylated, it releases beta-catenin in the cytosol. This now beta-catenin can be degraded, or it can translocate to the nucleus, where it can, for instance, lead to the expression of some target genes by associating with some transcription factors. And one of these target genes is REST. So to test whether mGluR5 had anything to do with the complex N-cadherin and beta-catenin, we have stimulated the receptor with the agonist or blocked with CTEP and measured the levels of N-cadherin phosphorylation. And what we observe is that when we stimulate the receptor with the HPG, we observe an increase in N-cadherin phosphorylation. And when we block mGluR5 with CTEP, we observe a decrease in N-cadherin phosphorylation. In terms of total beta-catenin, we don't observe any change following DHPG stimulation or CTEP blockage of the receptor. So this beta-catenin now release could be free to move to the nucleus and modulate gene expression. But how could mGluR5 modulate or phosphorylate N-cadherin? Well, it is well known that mGluR5 can activate many kinases, and our hypothesis is that mGluR5 could be leading to the phosphorylation or the activation of SERC kinase, which would lead to the phosphorylation of N-cadherin. So to test whether SERC activation would follow the same time frame as N-cadherin phosphorylation, we stimulated primary neuronal cultures with DHPG, or we blocked mGluR5 with CTEP for various times, and we measured the levels of SERC phosphorylation. And we observed an increase in SERC phosphorylation when neurons were stimulated with DHPG, and we also observed a decrease in SERC phosphorylation when mGluR5 was blocked with CTEP. And the time was quite similar to that of N-cadherin phosphorylation. And to further explore whether really mGluR5 and SERC activity could be leading to or modulating the complex beta-catenin and cadherin, we performed co-immunoprecipitation experiments where we IP beta-catenin and we did Western blots to determine the levels of N-cadherin. And these neurons were incubated with CTEP, which blocks mGluR5, as I said, or with a block of SERC. And as we can observe, when we block mGluR5 with CTEP, which means that we decrease N-cadherin phosphorylation, we see an increase in the interaction between beta-catenin and cadherin, showing that the receptor is important to modulate this complex. Also, if we block SERC with the specific inhibitor, we see an increase in the interaction between beta-catenin and cadherin, showing that mGluR5 and SERC are really important for the modulation of this complex. We then went on to perform some in vivo experiments. For that, we used the knock-in Q175 mice, as I said, a mouse model of Huntington's disease. And to our surprise, we could observe by Western blot that in the Q175 mice, there is an increase in SERC phosphorylation already at basal levels. And this increase in SERC phosphorylation went back to control levels when these mice were treated with CTEP, 2 milligrams per kilo for 12 weeks. Also, N-cadherin phosphorylation was increased in Q175 mice. And again, when we blocked mGluR5 with CTEP, we decreased the levels of N-cadherin phosphorylation. We did not observe any change in beta-catenin expression comparing wild type and Q175 mice. And also CTEP did not modify beta-catenin protein levels. And in terms of REST, when we blocked mGluR5 with CTEP in Q175 mice, we observed a decrease in REST expression, which was followed by an increase in SNAP-25 expression. So this data show that it seems like that indeed mGluR5 is important to modulate REST in vivo. And was interesting that we observed a more striking change in Q175 mice than in wild-type mice. We also tested whether mGluR5 could modulate REST in another mouse model of Huntington's disease, in this case, the transgenic BACHD mice. And for that, we did qPCR to measure REST mRNA levels, as well as SNAP-25 mRNA levels at 6 and 12 months of age. In wild-type, BACHD, mGluR5 knockout, and in the double mutant mice. And what we can observe is that REST expression is not changed in BACHD mice. And, however, the knockout of mGluR5 led to a decrease in REST expression regardless of the presence of mutant huntingtin. In terms of SNAP-25, we observed a decrease in BACHD, and that's expected, as I've mentioned before, that mutant huntingtin is unable to trap REST in the cytosol. But when we knock out mGluR5, we see a striking effect, an increase in SNAP-25 expression. So showing that in two different mouse models, mGluR5 can modulate REST expression. We also confirmed this data by Western blot, and we found the same results. So with this data, what we have shown is that in Huntington's disease, SERC phosphorylation is increased, as well as N-cadherin phosphorylation. And as I mentioned before, REST is released from the cytosol as mutant huntingtin is unable to trap REST in the cytosol. But when we block mGluR5 with CTEP, we see a decrease in SERC phosphorylation, a decrease in N-cadherin phosphorylation, and also a decrease in REST expression, which then can normalize the translocation of REST to the cytosol, to the nucleus. We then went on to look into CREB. As I mentioned before, CREB can be modulated by mutant huntingtin, and we already knew that CREB could also be phosphorylated by mGluR5. So mGluR5 can phosphorylate this transcription factor through the activation of different kinases, such as ERK, kinase 4, and adenylyl cyclase. And when CREB is phosphorylated and it's bound to CBP, it can lead to the transcription of many genes. And we therefore decided to measure CREB phosphorylation levels in these cross mice. And what we could observe is that in BACHD and mGluR5 knockout mice, as well as in the double mutant mice, we observed a decrease in CREB phosphorylation levels. When we look into two of the transcripts, CREB-dependent transcripts, such as BDNF, which is an important brain factor, and syntaxin, which is a snare protein, so it's quite important for neurotransmission. We could observe a decrease in the expression of BDNF and syntaxin in BACHD and mGluR5 knockout mice, and we observed additive effect between mGluR5 knockout and mutant huntingtin, as the double mutant exhibit decreased levels of BDNF and syntaxin mRNA levels compared to BACHD. As syntaxin is widely used as a presynaptic marker, we decided to look into SNAP-25 mRNA levels. So PSD95 is very important for the assembly of the postsynaptic proteins. So it's present in the postsynaptic site. And we then measure the levels of PSD95, and we observe that mutant huntingtin does not change PSD95 expression. However, mGluR5 knockout decreased PSD95 expression regardless of the presence of mutant huntingtin. And this was quite interesting to us because it was not known that mGluR5 could modulate PSD95 expression. And we believe that this modulation is not through CREB or REST, as we do not see any change in PSD95 expression in BACHD mice. And we decided to investigate a bit further this modulation of PSD95 expression. So we look into different brain regions, so brain regions that express mGluR5, such as hippocampus, striatum, cortex. If we knock out mGluR5, we observe a decrease in PSD95 expression. However, in the cerebellum, which does not express mGluR5, we do not observe any change in PSD95 mRNA levels. We tried to replicate this data using primary neuronal cultures from mouse embryos, and we blocked mGluR5 with CTEP for various times. However, we were unable to see any change in PSD95 mRNA levels. We went on and we also employed neurons from human iPSCs, or induced pluripotent stem cells. I'm sure that Stevens will talk more about these cells later on. And we blocked mGluR5 with CTEP in these neurons, and again, we did not observe any change in PSD95 expression. So we thought that maybe astrocytes would be needed in this situation. So what we have done is that we used astrocytes, also derived from human iPSC cells, and we blocked mGluR5. It is known that mGluR5 is expressed in astrocytes, so we blocked mGluR5 with CTEP, and we added the conditioned media to the neurons, but again, the levels of PSD95 expression were unchanged. So we decided to employ another culture model, the neurospheres. So the neurospheres are 3D cultures that are derived from also human iPSC cells, and they have not only neurons here labeled with MAP2 in green, but also astrocytes labeled in red with GFAP. And when they are mature, they also express mGluR5 here labeled with a specific anti-mGluR5 antibody. And now, when we block mGluR5 with CTEP, we observe a decrease in PSD95 expression in these cells. So we believe that somehow blockage of mGluR5 in astrocytes leads to the alteration of some factor or that is not released, maybe trapped in the membrane, and this factor that we are yet to identify changes PSD95 expression in neurons. But we still have to do more experiments to really clarify this cell signaling pathway. But as we observe change in syntaxin and PSD95 expression, we decided to look into the spines. For that, we employed a very old technique in neuroscience, the Golgi-Cox technique to impregnate neurons. So then we could image dendrites, and here we have a Z-series of a dendrite, and we can now reconstruct these images, and we can not only count the number of spines, but we can also count examine the level of maturation of these spines, because spines can be classified in mature, such as stubby, mushroom, and branched, or they can be immature, such as philopodia, long, and thin. And here is an example of a dendrite of a wild type and BACHD mice treated with vehicle. And we can already observe that there is a decrease in the number of spines in BACHD mice. And here is after we treat these mice with VU0509551, which is a positive allosteric modulator of mGluR5. And this is only a seven-day treatment. And when we look at the type of spines, we can observe that VU551 has quite a striking effect on mushroom type of spines. So here is the blue bar in this graph. And we can see that in both wild type and BACHD mice, we see an increase in the number of mushroom spines. So when we look and quantify the number of spines, the total number of spines, indeed, BACHD mice has less spines than wild type mice. And when we treat these mouse with the positive allosteric modulator of mGluR5, now we don't see a difference comparing wild type and BACHD mice. But again, the major effect, we believe that's in terms of the maturation of the spines. Also, when we look into the number of spines in the cross mice, we can see that there's a decrease in the number of spines in BACHD, mGluR5 knockout, and double mutant. The decrease in the number of spines in mGluR5 knockout was quite a surprise to us. So the knockout of the receptor, here we have 12-month-old mice. So the knockout of the receptor can lead to a decrease in the number of spines, which was surprising to us. And then we decided to look into the number of neurons in the brain of these mice. And here we have slices that were labeled with antibody that binds to NEDD4L, which is a specific neuronal marker. And we have slices from cortex, hippocampus, and striatum. The example I'm showing here is just the striatum, but you can see in the graph that the number of neurons is decreased in BACHD, mGluR5 knockout, and double mutant. So again, we observed the decrease in the number of neurons in mGluR5 knockout mice at 12 months of age. And this data is important because many studies suggest that the blockage of mGluR5 with CTEP or other drugs could be an option to treat neurodegeneration. But this data suggests to us that chronic treatment blocking mGluR5 could lead to spine loss and neuronal loss. So we have to be very careful when we propose this type of therapy. And I guess this data can illustrate how complicated it is to develop drugs to treat neurodegeneration. That might be the reason why we still don't have a disease-modifying drug to treat neurodegeneration. And we believe that perhaps by studying the cell signaling pathways involved and activated by receptors, you can comprehend better how this receptor can be activated or blocked. And maybe by using biased drugs, that could be an option to develop disease-modifying drugs to treat neurodegeneration. Just to summarize the data, so what I have shown here today is that, and this was shown before, mutant huntingtin traps CDP in the cytosol, so the level of CREB transcripts are decreasing in Huntington's disease, and mutant huntingtin is unable to trap Rett, so Rett moves to the nucleus and can repress gene expression. If we activate mGluR5, we now increase CREB phosphorylation, so we normalize the levels of CREB-dependent transcripts. But in the case of Rett, what happens is an increase in Rett expression. So I have now more Rett being translocated to the nucleus, which could lead to further repression. So an additive effect between mGluR5 and mutant huntingtin. I would like to thank my lab members, especially those that were directly involved by producing the data I've shown here today. Including the PhD students, Jéssica Mabelli, Antoniana Gonçalves, and also Pablo Cardoso. I also would like to thank my collaborators at the UFMG and also abroad, and also the funding agencies. Thank you.
Okay, so am I back? Yeah, I'm back. So thank you, Fabiola. Well, it was like a really nice presentation. There were people asking for your CV to see more about your work on the YouTube. So first, I would like to open questions from our guests here on the Zoom meeting. If you guys want to like turn on your cameras and microphones and ask questions, just pop in. So I'm going to start with asking questions then. I really enjoyed like your data. I know a little bit of it because of the lab, and I've always wondered why we see those data with neurospheres. Like when you apply drugs, you see a reduction in the PSD95 mRNA. So I was always wondering why we see those effects in astrocytes and not in neurons. And if you could clarify a little bit that on your hypothesis of what the reasons why that happened.
Thank you, Pablo. Well, so I believe that we need in cultures like neurons and astrocytes in close proximity in order to have that modulation. And I think that the next experiment should be maybe using astrocytes from knockout, mGluR5 knockouts, or maybe neurons from mGluR5 knockouts, or find some way to knock out the receptor, only one type of cell, to try to better comprehend how this whole modulation starts. But I do believe that the modulation starts in astrocytes, as mGluR5 is expressed in astrocytes, and then PSD95 is expressed in neurons, so maybe there is some factor that's being produced in astrocytes and modulate the expression of PSD95 in neurons.
I can ask a question. I'm James Ellis from Toronto, Canada, and will be speaking next. But Fabiola, what an outstanding talk about mGluR5 and DHPG. It's something that I'll describe a little bit in my talk, but you know far more about this pathway than I do. It seems incredibly complex with, you know, transcriptional changes, translational changes, all sorts of intermeshing pathways. Is there an angle that you think is going to be most relevant for the Parkinson's and perhaps leading towards a translational outcome?
Well, I bet on positive allosteric modulators. So I have Jeffrey Cohn at Vanderbilt University. I don't know if you know him. He is very good at producing many different types of allosteric modulators for many receptors, and he also works with mGluR5. And these positive allosteric modulators are quite versatile, and I believe that if we do dissect the cell signaling pathways, we could find a drug that perhaps would activate one cell signaling pathway and not other. And maybe by stabilizing some conformation of the receptor, that would facilitate the interaction of the receptor with other proteins. So in the lab, we're also looking to other protein partners for mGluR5 to try to dissect the cell signaling pathways. But I agree with you, they are quite complicated. So it's hard to know everything.
Yeah, no kidding. Thank you.
Thank you, James.
So thank you, Fabiola. People in YouTube are saying that was a really nice talk and they enjoyed it. So moving to the next speaker, we'll have James. Tiago, do you want to introduce him?
Yeah, thanks, Pablo, for everything. I'm Tiago. I'm a PhD student here at the Medical College of Wisconsin, and it is my honor to introduce Dr. James Ellis. Dr. Ellis has a bachelor degree in microbiology and immunology by McGill University, up there in Canada, I believe, and a PhD in medical genetics by the University of Toronto. He's now the professor of the molecular genetics at the University of Toronto, where he holds a senior position at the Hospital for Sick Children, also in Toronto. His main research focuses on discovering the basic mechanisms that control gene expression and epigenetic reprogramming using iPSCs, or reduced pluripotent stem cells, mainly to model neurodevelopmental disorders such as Rett Syndrome and Autism.
Okay, shall I take over?
Yes, James.
Okay. All right, I think the screen share should be live. Can you see it, Pablo?
Yeah, it's live, yeah.
Okay. I just have an overlapping set of speakers. Let me just see if I can change my okay, I just had a thumbnail sitting on top of my slide, so I'll get back to this laser pointer. All right, thank you very much, Tiago, for that introduction. Welcome, everyone, from your neighbor far, far to the north. I've not had a lot of interactions with Brazil until the last 8 years or so, and those interactions since then have been very, very positive. I've been down and visited Rio and Sao Paulo and Stevens's lab, and interacted with Pablo in my lab, and have a couple of research associates in my group. Some of the work of theirs I'll describe, and they really are the foundation of the group. Also, I think the order of the speakers has been spectacular. Fabiola's talk was really informative for me. I'll also start by speaking on mGluR5 using DHBG in autism spectrum disorder that's related to SHANK2 mutations. So that'll be one story. And then I'll switch over to translational regulation in Rett syndrome and a little bit on network activity as it shows up here. And if there's time, so please cut me off if I'm going a little long, I might be able to show some unpublished results as well on transcriptional rate and half-life post-transcriptional regulation. So on this first slide here, This is just to remind people about autism and that it's really related to neuronal dysfunction. So over here we have just the nice images that were also shown by Fabiola with the interactions between two neurons and a synapse between the axon and dendrite here. And this is an excitatory synapse here with glutamate being the neurotransmitter, various receptors, including the glutamate receptors that she was talking about. And as you can see, some of the signaling of which she addressed in much more detail than I'm showing here, going through PSD95, goes through some proteins called shanks. And what I'll describe first of all is a paper that we published on SHANK2 IPS-derived phenotyping. So IPS cells were mentioned in the earlier talk. This is just an overall summary that you can take somatic cells and reprogram them with 4 transcription factors to make induced pluripotent stem cells. These are pluripotent and they can differentiate into essentially any cell type. We tend to make neural progenitor cells from them and then differentiate them into neurons. You can do disease modeling in a dish or drug screening, and eventually toxicity as well for new drugs. And other people use cell therapies, which is not a particular approach for our lab. We're focused on disease modeling. So the 2 SHANK2 ASD cases that we studied are shown here with family 1 and family 2. You can see that they both have autism. They have issues with their IQ levels and their language measures. So the proband in each case is quite restricted in their behavioral abilities. So with family 1, this is a heterozygous stop mutation in SHANK2. In the proband, we made IPS cells, 3 IPS cells from this individual and an IPS cell line from each of the parents. With family 2, it's a deletion, a large deletion, so essentially another null, but as a heterozygous, again, 3 lines compared to 2 lines from the father. We also have an unrelated healthy control where we did a gene edit and did a homozygous knockout. So in this case, there'd be no SHANK2. And we took this point mutation here and gene corrected it as well. So we have another isogenic comparison. So we've got 2 isogenic pairs. When we got 2 family combinations. When we differentiate these through neural progenitor cells and into neurons, what you can see is the control cells are expressing normal amounts of SHANK2. The probands are expressing about half, and that homozygous knockout is not expressing SHANK2 at all. When we started to evaluate the phenotype of the neurons, what we do is we make neural progenitor cells, we plate them out onto mouse astrocytes and differentiate them for about up to 12 weeks. I think for the SHANK2 story, it's been primarily 9 weeks, but sometimes you can go shorter than that. And a neuronal network would form if you stain for synapsin 1 and MAP2. MAP2 shows the dendrites, synapsin 1 shows the synapses. You can then start to look at connectivity. But one of the problems that we ran into were the different lines differentiated with different efficiencies into neurons. So you can see this line here, the density is actually very low, whereas this line is very high. So if you started to look at the number of connections, cells in this high density compared to cells in this low density, it's clearly going to be high, and that higher here than there. And that leads to a lot of variability, which is summarized here with cell density varies between the cultures. And this leads to this variability in neuronal connectivity. So we came up with a highly controlled assay that we called SPARCON to detect dendrite morphology and electrophysiology of differentially marked neurons in the same well. What's being done here is we take 96% of a control neuron population. And so every well has a lawn of these control cells. And then we can take other cells that are either mutant or wild type and labeled with a chem kinase 2 promoter driving either a red or green fluorescent protein. So this will express in excitatory neurons. And by co-culturing them, you have some red neurons in here and some green neurons in here. You can see if the mutant or the wild type, which one is more or less active than the other. And so that you do this by differentiating the neural progenitor cells into neurons. We would label either the wild type, the control, or the mutant in green or red. And we do this blind so that the people who are looking at the data don't know what to expect. We then mix and reseed with this 2 to 96% ratio, mature them for another 5 weeks, and then stain to look at synapse number, as is shown up here, to trace morphology and synapses as shown in here, and electrophysiology. So these are 2 cells, one is mutant, one is wild type, in the same wall, and you can see, is this more or less complex than that, and normalize everything to the control. When we do that, these are the mutants, the SHANK2 mutants here, and their controls. So this is the point mutation compared to the parent, the deletion compared to that same parent and that family, the homozygous knockout to its isogenic parental cell line, and then we took this point mutation and corrected it through CRISPR gene editing back to the wild type. And you can see, just looking at the morphology, that these are much more complex looking than their isogenic controls. And when you go through and do the statistics, you can see that, yes, indeed, all of them have increased dendrite length. And here we've grown the mutant cells on a control line of neurons, but it was pointed out that in vivo, the mutant neurons would not be surrounded by control neurons, so we redid it with them growing on unlabeled mutant cells, and you still see this phenotype. We also looked at synapse numbers. I'm not going to show you that data, but dendrite length is up, synapse numbers are up, and we focused on the dendrite length. So SHANK3 had been previously reported by Ricardo Dalmash's lab. And in that case, they see that there's less connectivity and the dendrites are smaller. And they were able to rescue with BDNF and IGF-1 going through this mTOR pathway, and mTOR regulates translation. So this leads to improved function of SHANK3 neurons. And although this is making cells larger and more active, we were asked to test these same compounds in the SHANK2s. And what we found when we treat with IGF-1, BDNF, or TG003, what you see is IGF-1 does in fact make wild-type cells larger. These are the point mutants that are already larger, and they become even larger with IGF-1. And it's the same thing with the other 2 as well. So the compounds known to rescue SHANK3 phenotypes actually make the dendrite length longer in the SHANK2 neurons, and are not appropriate for considering potential treatment options. We also collaborated with an electrophysiologist in our institution, Mike Salter, and his research associate, Wenbo Zhang, who did patch clamp recordings. What you can see here is down below the mutants compared to their relevant controls. And these are excitatory postsynaptic currents. So you can see the frequency of them is much higher, and also the amplitude in many cases is higher than the amplitude shown here. So this is again done on these differentially marked cells within the same well, and you can see that in each case the EPSC frequency is up, even when they're grown on the mutant lawn of neurons, and the amplitude is also up, with one exception here being this here. So the frequency of the activity is increased really quite dramatically in the SHANK2 neurons, up to almost 8-fold. To look at mechanism, we did RNA-seq. We sorted the neurons because they'd been grown on astrocytes. And what I really want to focus on is this 9-week time point here, looking at synaptic markers. You can't see much from the count numbers here, other than that's primarily glutamate kind of genes that are being expressed. But if you look down here at the log fold changes, you can see that at the 9-week time point, when the electrophysiology and morphology assays were done, a lot of these synaptic genes are actually upregulated. And 2 of them that you can see here, GRIM5 and GRIM1, are those mGluR receptors. When we compare the overall transcription changes, to other reported ASD modules that are affected in idiopathic autism, you can see that at the 9-week time point, we see a lot of common changes that have been described in a couple of papers from genetics groups. So here is where we get a little bit of a link to the mGluR story, is that along with the BDNF treatment, we did a chronic DHBG treatment, and DHBG acts by interacting with mGluR, and one of the things it does is it signals through SHANK2. So if SHANK2 is defective, and here's HOMO over here, maybe we would see some changes. This was an idea that was put forward by one of my graduate students to just try DHBG for a 5-week chronic treatment. And what you can see is DHBG on the control cells has no effect. This is on total dendrite length. The point mutation again has longer dendrites. And when we treat with DHBG for 5 weeks, now the length phenotype is corrected. And this is in a situation where the mGluR receptors are presumably active and more abundant. So the conclusions from this SHANK2 work is that SHANK2 neurons from ASD subjects are hyperconnected, and this was verified by gene editing. The isogenic SHANK2 transcriptome is perturbed in these ASD gene modules, and dendrite length is rescued by DHBG treatment, implicating this mGluR activity-dependent pathways. And we've been more focused on translation. We haven't really done much related to transcriptional activity-dependent transcription as described by Fabiola. Now, I want to point out that not all ASD neurons or individuals are going to have these hyperconnected phenotype. The SHANK3 is hypoconnected, as reported by Dolmash's group. And we had a paper that just came out earlier this year on a long non-coding RNA that is actually highly mutated in autism individuals. And in this case, they're hypoconnected. So just putting that balance, either too much or too little connectivity seems to be able to contribute to autism. All right, so I'll change topics to Rett syndrome. It's another neurodevelopmental disorder that affects girls. It affects girls because the mutations are in the MECP2 gene, which is on the X chromosome. Girls develop normally for a year or two, but then they regress and they lose language and have another a number of other serious phenotypes. So MECP2 is a methyl CpG binding protein 2, and its traditionally accepted role is as a transcriptional modulator, usually repressing genes, but it's actually being reported to activate genes by interacting with CREB. In mouse models, a number of features have been identified about morphology and synapse numbers and synaptic transmission. And most of these have been reproduced in Rett syndrome iPS cell lines by Alison Moatri, a Brazilian scientist in San Diego, and ourselves and others. But coming back to MECP2, it's also known that MECP2 impacts translation through the mTOR pathway that I mentioned earlier. It also regulates processing of microRNAs, which might impact half-life of mRNAs. And therefore, we wanted to follow up on the translational, on the post-transcriptional angle in Rett syndrome. So what was done, this is the work from David Rodrigues, who's a Brazilian research associate, and the work that was published earlier this year, is to look at translational regulation using a technique called TRAP-seq. And I'll just give you the final message first, which is we identified ubiquitin pathway defects in Rett syndrome neurons that affect the cell surface proteome. So the way the experiment is done is that you express a fusion protein between a ribosomal protein and EGFP in cells, and we went into iPS cells, neural progenitor cells, and neurons. what happens is the GFP tagged ribosomes will be bound to some transcripts that are being translated, but not to other transcripts that are not being translated. So the way you do it is you have your cell lysate, you split it in two to do RNA-seq to identify the transcriptome of all the transcripts, including the ones that are not bound by ribosomes. And trap-seq, you're sequencing the transcripts that are being pulled down through the GFP tag on the ribosome. So this gives you the translatome. And the overall message is that when we look in Rett syndrome neurons, this RE stands for ribosomal engagement. Overall, there's a reduced translation, global translation in Rett syndrome neurons, but there's also a high number of genes that are translated specifically to a lower degree than in wild type. And the ones that are most affected are these 5 here, which are all E3 ubiquitin ligases, leading to reduced ubiquitination in these Rett syndrome neurons. So I'll run through the data. This is comparing the ribosomal engagement between Rett syndrome neurons and wild type neurons. You can see the most highly engaged in ribosomes are these 5 shown here, which are all in the same family of E3 ubiquitin ligases. And I'm going to be focusing on this because it's off on the left side. It means they're more engaged in the wild types than they are in the Rett. They're still engaged to high levels in the Rett, but in a differential manner, it's the most extreme. When David did Western blots, the Rett syndrome cells were null, so they are not expressing MECP2, and I'll just hop straight to this NEDD4L. You can see that there's barely any protein in here. And the other ones that are affected up here do have some reductions in the protein as well. So if you compare the RNA levels using qRT-PCR, you can see that all of these genes, the RNA is unchanged. So you wouldn't pick this up in an RNA-seq kind of experiment, but with trap-seq you do. And NEDD4L has this huge difference in the protein levels, and the others do have statistically different differences in the protein levels, with the exception of this itch1. So is NEDD4L of any relevance to the biology of what we know with Rett syndrome? This was a paper from 6 years ago in PNAS showing that NEDD4 acts as a downstream target of the mTOR pathway, and it promotes neurite growth. So if you're making less NEDD4L, you might be reducing neurite length, which is one of the known phenotypes of Rett syndrome. So it makes sense biologically. What could those E3 ubiquitin ligases be doing? The E3 ones are the ones that recognize the target proteins and transfer ubiquitin on them. And some of the known E3 targets that are already being reported are these potassium channels and neuroreceptor channels here. So normally they get ubiquitinated and degraded in the proteasome. If NEDD4L is less abundant, you'd see less of this ubiquitination, less degradation, and you'd actually expect these proteins on the cell surface, which are functionally really relevant to synaptic connectivity, you'd expect them to go up. And when you do these Western blots over here, you can see that in the Rett syndrome cells, the protein levels actually do go up, and quantified here. And again, none of this is revealed through RNA-seq or RNA, RT-PCR kinds of analyses, because the RNA is not actually changed. One of the other surprising things that we found with the trap-seq data is that long non-coding RNAs are categorized as non-coding because the only open reading frames that they have are less than 100 amino acids in length, and it was felt that small proteins like that, small peptides like that aren't genuine. So they're categorized as non-coding. But what you can see here, this is just looking at neuron or Rett syndrome neurons, you can see that mRNAs, which you'd expect to be making proteins, normalized around this ribosomal engagement level of 1, have, most of them are being translated. If you look at long non-coding RNAs that are intergenic, you see that a lot of them are not being translated, as you'd expect. But above this cutoff of about 0.8, which is where you see the mRNAs really being engaged, you can see that a lot of these long non-coding RNAs are actually engaged with ribosomes. And that this is quantified down here, where you can see that roughly a third of the long non-coding RNAs that we could see in each of these cell types are actually engaged with ribosomes really highly. And then it's been reported over the last couple of years that long non-coding RNAs often do encode micropeptides, and some of these micropeptides have been well described, especially in the heart context. And what we are doing here is looking at wild-type neurons compared to Rett syndrome neurons of long non-coding RNAs that we see engaged with the ribosomes. And you can see that a number of them are actually dysregulated in Rett syndrome. And these ones in red are the most significant ones where micropeptides have actually been discovered in other systems. So we're very curious about the potential role of these long non-coding RNA encoded micropeptides and the ubiquitination as well. Now, I mentioned that I'd talk about connectivity. This is the only slide that I've put in about connectivity. It's a paper that's on bioRxiv right now. And one of the things is if you've got, ubiquitination pathways or, mGluR pathways or micropeptides that you want to test their role and function, what is the most relevant thing to look at? And connectivity across neurons is one way to do it using what's called a multi-electrode array. So this is a well here. You can see 4 electrodes, but in each well that we use, there are 64 electrodes. Each of these lines going across is an electrode. And you can see that in wild-type control neurons, you get all of these neurons and electrodes firing at the same time. So you get what's called a network burst. And if you amplify that, you can look at the duration of the network burst. If we look at MECP2 null cells, you can see just looking at it directly that there's fewer of these network bursts and that the duration is much longer. And again, how we've quantified them is to look at the relative number of these normalized to the controls on the same plate. And you can see that, yes, at different weeks of development of the neurons, by the way, these are neurogenin 2 induced neurons, you can see that the network burst frequency is down and the network burst duration is up. And this is the kind of assay that once you describe these phenotypes, you can then use to test potential drugs. So the conclusions from the Rett syndrome work is that the neurons have defects in translational regulation that reduce E3-ubiquitin ligase levels, which in turn increase target proteins, and none of these correlate with the mRNA levels. That long non-coding RNAs are engaged with ribosomes and neurons and may encode micropeptides, and that Rett syndrome neurons have defects in circuitry with fewer network bursts and longer durations. Now, Pablo, if it's okay, I can show another 5 slides. I think I probably have time. Are you okay with that?
Go on.
Yeah? Okay.
Yep.
So because we've been interested in post-transcriptional regulation, I mentioned the translation work in the previous paper by David. We also wanted to look at RNA stability. And one way, and RNA stability is regulated by microRNAs, and microRNAs are known to be impacted in Rett syndrome. So one of the ways you can study RNA stability in Rett syndrome neurons is a technique called rate-seq. What rate-seq does is it incorporates 4SU over time, so half an hour, out to 24 hours. And you can compare your starting amount of RNA to the proportion that is labeled with 4SU to come up with a half-life, because you can see that in here what's happening is you get transcription, then RNA degradation starts to occur, and you hit an equilibrium up here where transcription is being accommodated by RNA stability, and that gives you the value that you get in RNA-seq data. Now, David had previously shown in a paper 4 years ago in Cell Reports that MECP2 has 2 3' UTR isoforms with different regulation, different half-lives. And when we did this rate-seq experiment, he could validate that, yes, those half-lives are what we had already shown to be the case for MECP2. And then he validated using QRT-PCR half-life experiments that an additional 7 that were found in the rate-seq experiments really did have altered half-life in the Rett syndrome neurons. So that's 7 candidate transcripts that were experimentally validated in these Rett syndrome neurons. And David had done the experimental work, and the computational work is done by Murat Mufti, a computational PhD student in my lab. But what we didn't realize at the time when we started this is that you can also calculate transcription rates in these neural progenitor cells relative to the iPS cells, or transcription rates in any of the cell types. So again, what you're doing is incorporating 4SU. After 30 minutes, you're looking at the labeled RNA. After an hour, you're looking at the labeled RNA. And most mRNAs have a longer half-life than just an hour. So what you're measuring here is the transcription rate. When you do this and you compare, what we're comparing is wild-type NPCs to wild-type iPS cells. If you look at housekeeping, this is the transcription rate of the housekeeping genes, it's essentially unchanged between iPS cells and neural progenitor cells. If you look at the progenitor genes, yes, transcription rates are going up, as you'd expect. And if you look at the iPS cells, the pluripotency genes are high in the iPS cells and they're going down in the NPCs. So this is giving you confidence that you actually are measuring relative transcription rates and that they do change as the cell types change. And the surprising result to us is that if you look at Rett syndrome, you get about 1,250 genes that shift steady state mRNA levels as detected by RNA-seq. But if you start to calculate the transcription rate and the half-lives, you see a different picture. So there, if you look at genes that are regulated only at the transcriptional rate level, you see about 36%. So this third of genes. If you look at genes that are purely regulated at the half-life level, you see this 12% over here. And then roughly half of the genes are influenced by both by changes in transcription rate and changes in half-life. And these steady state levels can also be buffered by compensating shifts in transcriptional rate and half-life, because if your transcription rate goes up, but your half-life goes down, you would then expect to see no change in steady state. And so your RNA-seq data says nothing's happened, but in reality, things have happened, they've just buffered each other. So I think this is my last data slide, and this is examples of genes that are regulated or buffered by transcription rate or half-life in Rett syndrome neurons. So WWC1 is purely regulated transcriptionally. You can see transcription rate is up about threefold. The steady state by RNA-seq is up about threefold, but half-life doesn't change. So that's purely transcription rate. And that's what most people would think would change in a Rett syndrome context because it modulates transcription. But if you look at stability, here's GRIA1. You can see that the transcription rate does not change at all in Rett syndrome versus wild type. But the half-life actually does change over here and can account for this change in steady state. And this is an example of buffering. ATRX is sometimes associated with autism. You can see the transcription rate goes up, down, the half-life goes up, and steady state analysis reveals no change. So if you do RNA-seq, you say ATRX is not changed, but in fact it is. It's just they've buffered and canceled each other out. So in conclusion, chromatin remodeling genes like Rett syndrome, but there's a lot of other chromatin remodeling genes that are in, that cause neurodevelopmental disorders. They may affect more than the transcription rate alone. RNA-seq detects the steady state. What I've shown you is trap-seq to look at ribosome engagement, and that there are actually more genes that are influenced at the ribosome engagement level than at the steady state level of RNA in Rett syndrome. And I've shown you that the ubiquitome changes in Rett syndrome, and that probably micropeptides also change. And there are proteomics approaches like diglide TMT or parallel reaction monitoring that can be used to detect these sort of changes and the impact of the protein on the cellular function. And I've also shown you that going upstream of the mRNA steady state, you get changes in both transcription rate and mRNA stability. Using rate-seq. We also have microRNA-seq to help try and explain this. And we're currently looking at these role of RNA binding proteins and microRNAs in mRNA stability. So overall, we're going to be finishing off this work hopefully in the next year and pursuing it. But from a therapeutic potential aspect, we want to identify the ubiquitome of the Rett neurons. to see what changes are happening at the ubiquitination level, and can we rescue this with compounds that are directed at ubiquitin E3 ligases, and also search for evidence that the long non-coding RNAs actually do encode micropeptides that are differentially expressed in the Rett syndrome neurons, and start looking to test their function in the Rett syndrome neurons, and the multi-electrode array could help address an assay to do that. So in acknowledgement, the people who did the bulk of the work here are in red: Kirill and Fraser for SHANK2, David and Murat for the TRAPseq, Rebecca for the MEAs. We got a lot of help from our technicians in making neurons. Wembo did patch clamp, and some of the translational calculations and RNA stability calculations were done in addition in Ben Blankos and Quade Morris's lab. And we have additional collaborators, including Alison Mwatri, who is Brazilian. These are the funding agencies, and these are the people in the group. And I'll just point out David, and thank you for your attention.
Hi, James.
Hi,
hi, I'm Bruna. Nice to meet you. Very great talk. I have a couple questions for you.
Yeah.
So I found it very interesting when you were showing kind of like opposite results in between SHANK2 and SHANK3. While like one gives you a hyperconnectivity, the other one seems to have like the opposite direction. And I both converge, at least in the level of causing autism or neurodevelopmental disorders. So I would like you to discuss a little bit more about that. And the other very interesting point that I found, and like although we already have suggestions about that, is that like not always everybody has been talking a lot about single cell RNA-seq and RNA-seq, but you may miss a lot in RNA-seq experiments. So I really think that, and I would like to know your opinion about it, that we might need techniques that may not be here yet, but like combining RNA level, protein level, and perhaps even more to really understand molecular basis of neurodevelopmental disorders like ASD, at least in the cellular level in vitro. So yeah, if you could talk a little bit more about these 2 points, very exciting. Thank you.
Usually I forget the second question, but I think I'll remember it this time, because
I can remind you.
The first question, you know, I'm going to have to be a little bit like the American debates and just stall, because I don't have a good answer. I mean, why SHANK2 would give you, you know, a hyperactive situation, and SHANK3 would give you a hypoactive is, you know, I think we just, I mean, we're not experts in the scaffolding function of the shanks. And so I guess it would imply that the different shanks are doing different things as scaffolding proteins. We do feel like SHANK2 is maybe involved in regulation of ARC, and ARC is involved in, you know, sort of synaptic plasticity and stabilizing synapse numbers, and maybe that kind of a process can help stabilize those synapses and maybe promote dendrite growth. But I mean, to be perfectly frank, I don't have an answer for you. It's too good a question. There's not a good answer that I have. For the single cell, yeah, with the RNA-seq and what gets missed with RNA-seq, I mean, everyone's acknowledged that, you know, post-transcriptional regulation is important. We all know about it. My own background was working on DNA regulatory elements during my postdoctoral fellowship that control beta globin gene transcription. And my perspective had always been, well, you get the RNA and everything else follows the RNA. Like, you know, yeah, these other things happen, but don't worry about it. And I never gave it any second thought until actually David arrived in the lab. And David had experience working, I think it was on Chagas disease and post-transcriptional regulation. And he noticed that there was a very large, well-conserved 3' UTR on MECP2, and wanted to look to see if that regulated post-transcriptional for MECP2. And I agreed that it was a sensible experiment. And he showed it, and he's developed gotten most of these techniques to work. and really opened my eyes. And I think it's important for trainees to realize this, is that you can change the direction of a field or a lab just by bringing in new ideas and making it happen. And David's been able to do that. And we got into iPS cells because we had a Japanese postdoc working on a pluripotency reporter. And he was testing it in ES cells just the time when Yamanaka reported. And so he said, I want to do reprogramming, and found the vectors on add gene. And, you know, by, I think it was early 2008, we had that mouse iPS cells growing. It was fantastic. And he did really well. So students can really contribute to the direction that a lab and a field goes to. And I agree completely that single cell RNA-seq and, you know, all of the chromatin work that gets done, single cell ATAC-seq, all of these things are really important. And, you know, I showed that, what was it, you know, almost, what would it be? Almost 90% of genes are influenced by transcription. So really important, but it doesn't correlate with protein levels. And that's been known for years and years and years. And so if what you're trying to do is manipulate proteins for therapeutic use or to look at pathways, you have to incorporate additional new technologies. So yeah, sorry that was such a long answer, but it was a great question, Bruna.
That was great, thank you. Any more questions?
I have one question for James. It's it's very interesting, your data regarding the transcription rates. And I'd like to know if you have considered to use like inhibitors of the histone deacetylases in your system to see if you're going to shift the transcription rate, for example, like antidepressants.
Yeah, that's a great question. We haven't actually considered focusing on the transcription side because we're mostly concentrating on the post-translational side, which is, you know, less well studied. But you're right, that would be a good proof that you're looking at transcription rate. I think I'm forgetting a control that we may have done to start addressing that. But that would be a really interesting experiment to look to see how do HATs impact transcription rates. I think it's really, you know, the rate-seq data is really important because, you know, things, you know, these chromatin remodeling complexes that are involved in disease, everyone just assume it changes transcription rate, but there's no data at all that it changes transcription rate. It's just RNA abundance is up. That's not transcription rate, and it has not actually been studied.
So we've got one question. Actually, this one came from WhatsApp instead of YouTube. So let me take myself in here. So it's more related to other cells in the central nervous system, like glial cells, because most of your data it's regarding some neurons, but the glial cells also modulate several important key aspects in the central nervous system. So have you gotten any look into glial cells in modulating different aspects of Rett syndrome? And if so, if they change anything, like, from example, let me check here, electrical stimulation, for instance, in the neurons.
Yeah, so another great question. Pablo, you remember that when you were in the lab, you were actually trying to do some astrocyte experiments. And so that was our first attempt to do that. And, you know, I was asking a lot for an undergrad student to make progress on that. And you gave it your best shot. But we do now have a PhD grad student working on making astrocytes from Rett syndrome. What we want to be able to do is to change our multi-electrode arrays so that now we can grow them on control or Rett syndrome human astrocytes rather than on the mouse astrocytes that we currently use. Other groups are succeeding in doing this. I mean, I think Fabiola mentioned that she used iPS-derived astrocytes in some of her experiments. So we're catching up, we're learning how to do this. And I think with the, so with the astrocytes, we're really curious about things like are there changes in the ubiquitination? Are there changes in the micropeptides? Do they start making factors that are important for normal neuronal maturation and function? So this is a really important angle that we're beginning to do, but we're nowhere near as accomplished in working with astrocytes as other groups. And certainly a number of other groups have already shown that astrocytes are really important for, if you take Rett syndrome astrocytes and you put on control neurons, that actually makes those control neurons less functional. So it's a really important field that we would like to get into.
So thanks, James. We are going to move to Kristen now, our next presenter. So Kristen has a bachelor degree in cell molecular biology by the University of Calgary. Am I right?
Yeah, in Calgary, that's correct.
Okay. So, and she also has a PhD degree in biological sciences by Harvard University. Currently, she's an associate professor at the departments of genetics and genomics, and neuroscience and psychiatry in the Icahn School of Medicine at Mount Sinai in New York. And her major research focus is in developing in vitro models for schizophrenia in order to identify novel insights into the molecular and cellular phenotypes of mental illness. Kristen is going to give us a talk that's entitled Using Stem Cells to Explore the Genetics Underlying Neuropsychiatric Diseases. So thank you so much for joining us, Kristen. I'm really glad that you accepted our invitation along with the other speakers. And so the screen is yours.
Thank you. I'm really excited to join everybody too. And it's been really fun to watch the other talks, and hopefully I can add a little bit more to that. So as Pablo noted, today I'll be talking to you about using stem cells to explore the genetics underlying neuropsychiatric disease. And I think the first question really is to back up to why. If you are a psychiatrist, there's understandably a lot of skepticism about what stem cells, what cells in a dish can teach us about a whole brain, and a class of disorders defined by circuit function and behavior. And so I think that our hope as a field is to really inform in two really necessary places. And the first is diagnosis. There are no biomarkers for psychiatric illness. There are no biopsies, and there is no genetic-based diagnosis. And so I think that if we could help move towards a blood test for schizophrenia or autism, something where you can diagnose a patient prior to symptom onset, then we not just improve diagnostics, but we extend the period of therapeutic intervention. And the second hope, of course, is that if we can understand the molecular mechanisms underlying disease risk and the cell types first affected, then this too might identify pathways for targeted intervention to either prevent or treat disease. And so, of course, what I'm really talking about here is precision medicine. The idea that the effects of variants or the interactions between those variants impact not just our disease risk, but our treatment response. And so I'll focus largely on schizophrenia. And for those of you who are less familiar with this disorder, what you need to know is that it's common. So it affects about 1% of people in every country on the planet, and that includes about 3 million Americans. It's really severe. So patients have a 1 in 10 chance of suicide once diagnosed, a 1 in 2 chance of drug and alcohol abuse problems. 9 out of 10 will not hold a full-time job after being diagnosed, and 1 in 5 will live on the streets. There is no cure for this disorder, but it is extremely heritable, with estimates approaching 85%, which makes it more heritable, for example, than things like alcoholism and BRCA1 breast cancer, which we classically think about as running in families. And so here we're graphing heritability on the y-axis and prevalence on the x. And what you can see is that autism, schizophrenia, and bipolar, they all cluster together as extremely heritable brain disorders. Now, how this heritability is reflected is actually still has a lot to deconvolve. You can imagine a lot of theoretical ways of how one might inherit a variant. And one of them is, of course, Mendelian variants. So these are very rare in the population, because if they were common, then all of us would have the disorder. But these variants have very large effect. In the case of 22q11, you are very, very unlikely to be a control if you carry this deletion, this copy number variant. It's very hard to identify rare variants of small effect, and it's very unusual to identify common variants of large effect. So the other major class of variants linked to psychiatric disease are common variants of very small effect. So here the effect sizes might be 1 or 2 extra% of risk for each variant carried. And to be honest, each of us might carry dozens of common risk variants, but patients might just carry a few more. And so the most recent published GWAS for schizophrenia is at 145 common variant loci. The unpublished PGC3 is at around 250 common variant loci. And we are still very far from saturating the curve. To greatly oversimplify 50 years of clinical research, what I think is important to know about schizophrenia is that the neurons in patients are less well connected. So here we're showing some imaging scans led by Judy Rappaport, where she's highlighting in red are the brain regions that are smaller in patient brains. Now it's important to note that patient brains are not smaller because cells are dying. They're smaller because the neurons themselves are smaller. They're known to have fewer dendrites and fewer synapses, fewer connections between them. But we still don't, these postmortem pathology studies don't tell us about what happened to lead up to disease. And we know very little about disease risk and progression. And that's just simply because there's not enough live human brain tissue for mechanistic studies of schizophrenia risk variants and drug discovery. And while mouse models were generally really ideal for this sort of purpose, I think in terms of human behavior, it's something that we often think of as uniquely human. And more than that, there are other limits to the model. First of which is just the brain. So I didn't train as a neuroscientist, but I always like to joke that even I can tell the difference between a mouse brain and a human brain, and I still have that ability when we control for size. In addition to the differences in behavior and brain structure between mice and human, there's differences in brain sequence, in DNA sequence. So mouse models are fantastic at revealing the complex interactions between genes, circuits, and behaviors, but they don't well capture the impact of those variants that you can't put into a mouse model. So a complex rare variant, a large C and B deletion, can be very difficult to engineer into a mouse, and non-coding common variants that underlie most of the risks for psychiatric disease are generally not conserved between rodents and humans, and so again are very difficult to model in animals. And so this is why we've been pushing forward into using stem cells as an additional, not alternative, just an additional model to study psychiatric risk. And so really briefly, as the other speakers have already really well described, the idea is that you would take skin or blood cells from patients and controls, reprogram those to stem cells, and differentiate or induce those into neurons. And so when we first established this, and I like to show this slide, even though it's really, really old, because it always reminds me to make one really important point, which is at this level of magnification, there's no obvious differences between patient neurons and control neurons. And that's really, really important because patients walk and talk and breathe just like you and I. And so I want to stress here that these phenotypes are very subtle. We're looking for small effects. Before we talk about comparing case and control derived neurons, it's again important to ask to what part of the brain and to what stage of human brain development are the cells that we're making most similar. We first did this analysis in to be honest, 2013 or 14 by microarray of 4 control neural progenitor cells and 4 control neuron donors relative to the Allen brainspan microarray data set at the time. And what you can see in black is that our stem cell derived neural progenitor cells look most like first trimester fetal tissue. And even our neurons look most like second trimester fetal tissue. And this was at the time, I think, really disappointing because what we were hoping to say was that we were modeling a disease way over here on the right side of the graph. And of course, it's clear now that we are not. These are not disease in a dish models. These are disease risk in a dish models. We're modeling predisposition to schizophrenia with stem cell derived neurons. More recently, a collaborative of mine, Gabe Hoffman, went back and repeated this analysis on a much larger data set. So here he's got hundreds of brains from the Common Mind Consortium, the Allen Brainspan collection, the GTEx collection, as well as all of the IPS-derived neural progenitor cells and neurons that my lab had generated to date, that Tracy Young-Pierce's lab at Harvard had generated, and Gu Li Ming and Hong Jun's lab at Johns Hopkins at the time had generated. And so here are all the brain samples clustered together. In darker blue are the fetal samples. And in green are the neural progenitor cells from all three of our labs. And in orange are the neurons. And so what you can see is that when the neurons overlap with human brain tissue, they overlap with fetal tissue, again, consistent with the earlier analysis. That being said, I do think that we have been able to detect some differences in neurons from cases versus controls. This again is a really early analysis looking at postsynaptic marker PSE95 levels that are decreased in 4 patients. And I show this only because it was quickly replicated by a second group in Israel, which using the exact same assay, but 3 independent patients on a different continent were able to reproduce the same phenotype, which I like to think is the first bicontinental replication of a complex genetic phenotype in brain disease. At the time that we published this work, we actually were not able to show functional differences in synaptic activity, but I left behind the stem cells when I finished my postdoc, and Diana Yu carried this project forward. We had speculated originally that the reason we didn't see deficits was that the neurons were too heterogeneous. They were in a mixture of glutamatergic and GABAergic neurons. So Diana really cleverly found a way to just record synaptic activity from PROX1 positive glutamatergic neurons. And when she did so, she was then able to resolve deficits in the frequency and amplitude of excitatory synapses. And then one of the first phenotypes we found when I started my own lab was not in the neurons, but in the immature neural progenitor cells, the cells that will ultimately become neurons. And we were able to show that they migrated less well than the control NPCs. Not to say, of course, that we think cells are migrating improperly in patient brains, but perhaps to model the model. The cells that Diana were recording were 3 months old. And the cells that we assayed here were 2 days old. And so sometimes, but not always, a reasonably fast assay can be more scalable and useful than a slow and sensitive one. But the question then altogether is why? Why are these neurons, why do they seem to have different amounts of synaptic protein? Why are they firing differently? Why are they migrating differently? And do the molecular causes underlying these in vitro phenotypes teach us anything about schizophrenia? Do they represent the same genes as the genetic risk factors for schizophrenia? So this was the first undertaking when we established the lab. We took again neural progenitor cells from 6 controls and 4 patients, and we threw the kitchen sink at it. We had no idea what the mechanism was driving these phenotypic differences. So we started with microRNA profiling and RNA-seq. And so here's the results of the microRNA profiling. These are direct measurements of differences in microRNAs. And what we learned from this is that microRNA 9 is highly expressed in human neural progenitor cells, and that is also the most perturbed high or medium abundance microRNA in patient-derived neurons. Independently from our RNA-seq, my collaborator Gong Feng was imputing differences in microRNA levels by looking at the gene targets that were changing in the RNA-seq from these samples. And so here again, he imputed that miR9 was the microRNA driving the differences in gene expression. And so we looked up miR9 in PubMed, and we learned at the time that had been previously associated with neural differentiation and neural migration. And so we cloned it into some retrovirus overexpression vectors, and we were able to show that decrease in migration that we had observed in patient-derived neural progenitor cells was ameliorated by overexpressing miR9, and it was recapitulated by knocking down miR9 in control NPCs. We then read and replicated this in a second cohort. So this is the childhood onset schizophrenia cohort recruited by Judy Rappaport. And so here I'm showing you the normalized microRNA expression across both cohorts together. And I'm doing this to help me make 2 points here. First is that all patient neural progenitor cells had microRNA 9 levels less than the median value of controls. But I want you to look carefully and see that miR9 levels were not diagnostic. You could not tell who was a patient who was a control by miR9 levels alone. However, this statistically significant difference between the groups was driven by a subset of the NPCs. So about 50% of the patient NPCs had values less than the bottom quartile of controls. And so what you're really seeing here is a subsetting effect. And in this particular case, we had a large enough subset and a large enough deficit that we were able to see it in full case control comparisons. But I think moving forward to the second and third and fourth differentially expressed microRNA or gene, it gets harder and harder across these idiopathic cohorts. We were really lucky at the same time, the PGC2 came out, and a meta-analysis of all the loci linked to schizophrenia in the PGC2 identified 2 microRNAs with targets whose sites were enriched in the PGC2. miR-137 had already been associated with schizophrenia, and miR-9 was added to the list. And so this was a nice independent validation. I note, of course, here that this meta-analysis took one computational grad student 6 months to complete, and our if-space analysis took the lab 5 years and an entire R01 to undertake. And so this really taught me the lesson not to try to compete with the geneticists, but to work with them. And so that's really when the lab pivoted. We stopped trying to discover the mechanisms underlying these deficits on our own, and we started working really closely with geneticists. So here we're going to talk about applying stem cells to discover and functionally validate the impact of common variants. This is the most recent schizophrenia genome-wide association study that's been published. The red line indicates genome-wide significance, and there are 145 loci over that line. Now, in 2015 or 16, when this first came out, or when these data started to come back, when we wanted to validate one of them to show that one single nucleotide polymorphism was sufficient to change gene expression and maybe change neural function, I didn't know where to start. As a stem cell biologist, my instinct was to go after this really, really tall one. And I was really lucky to work with some really smart friends who kept me from that direction. And instead, they pointed me here to this loci containing the furin gene. And something, in fact, happens at this loci that doesn't happen at any of the 144 other genome-wide significant loci in this GWAS. So at this loci, what I'm showing you here on the y-axis is the same y-axis as the larger GWAS, and on the x is the brain EQTL. So the significance of an individual SNP to regulate expression of furin is being plotted against the significance of an individual SNP with schizophrenia risk. And what you see here is one SNP in the top right-hand corner. The very same SNP, RS4702, is both the most significant for schizophrenia risk and also the most significant to regulate furin expression. In fact, it lies in the 3' UTR of the furin gene. Now, most of the time, what you see is not this clear. And so this is the second best loci in the GWAS. And so here you have SNAP-91, and what you have is about 30 SNPs, all linked to schizophrenia risk and also linked to expression of the coding gene nearby, SNAP-91. And what we don't know is if one SNP in this cluster is responsible for the entire effect, or if each of these 30 SNPs is contributing 3% of the risk at this specific loci. So looking at furin, and this was work led by a really talented and I gotta say really brave postdoc in the lab, Nadine Shroud. So while on the previous slide, I showed you the best data related to furin and RS4702, here's the flip side of that same data set. So here we're plotting for any individual brain, the expression of furin relative to the genotype of the original donor at this loci. And while the effect is significant, the error bars here are extremely large. And this is because, of course, each of these brains came from a different person. And these people varied at many, many loci, not just RS4702. They died at different ages of different causes. Half of them had psychiatric disease and half did not. Half were on antipsychotics, the others were not. They had different histories of drug and alcohol abuse. And so overall, there were many, many things separating these brains. And our hope, our hypothesis, was that if Nadine did the experiment properly in a dish where she can control every single aspect, that the effect size would be the same, but the standard deviation between experiments would be much reduced. The idea here being she would use CRISPR technologies to edit a single non-coding SNP, here RS4702 from AA to GG. This actually took her a couple of years to do. It's technically very difficult because she couldn't edit the PAM site downstream. But when she achieved it, what she was able to find was actually, to be honest, better than we could have hoped. So with nothing different between these IPS lines, except for their genotype at this single SNP to our knowledge, allelic conversion from AA to GG significantly reduced furin expression in glutamatergic neurons. And as I mentioned, this is a 3' UTR SNP that's found in the binding site of a microRNA, miR-338. And inhibiting this microRNA actually obliterated the effect. And so it was a microRNA-dependent allelic conversion regulating the expression of the gene. GG neurons have reduced neurite length, and they have different firing patterns than AA neurons. However, for many of these other SNPs, CRISPR editing is just not a good strategy. And so here, led by Sukman Ho, we applied CRISPR activation and CRISPR inhibition to affect relatively modest changes of these genes linked to schizophrenia risk in the disease predicted directions. So here's the RNA-seq for reciprocal perturbations of SNAP-91 and tSNARE. What you can see is that the gene with the largest change is the target gene, but there are in fact other genes changing downstream. And those other genes are enriched for brain and synaptic gene sets. Reciprocal perturbations of SNAP-91 and tSNARE are sufficient to change the density of synaptic proteins, and they're sufficient to alter the excitatory synaptic activity by electrophysiology. So here, increasing SNAP-91 increases synaptic activity, and decreasing it decreases synaptic activity. But again, we are modulating just one schizophrenia risk gene at a time, and that's not how it works in disease. And so we asked what happens if we perturb schizophrenia risk genes in combination. And so again, Suk and Nadine teamed up to take this on. They conducted RNA-seq of single gene perturbations of 4 top schizophrenia EQTL risk genes. And then Nadine summed the individual RNA-seq profiles to generate an expected additive model of what she thought we might see by RNA-seq if we were to manipulate these 4 genes at once. And then of course, side by side, we actually did the experiment. And we asked gene by gene, how good was the expected additive model? And so we called the model good if there was no difference between expectation and measured. And then we called it synergy if the genes were markedly up or down regulated. And so it turns out most of the time, the model was really good. 82% of genes changed about as we predicted, but 7% of the time, genes were more down than expected. And those genes that were more down were enriched for all the major neurotransmitter release categories. And 11% of the time, genes were more up than expected. And the genes that were more up were enriched for both the rare and common variant genes associated with schizophrenia risk. And so I think this is a really interesting proof of concept, saying that there's emergent biology when you think about the context of genetic risk factors. So it's really important to think not just about one gene at a time, but understanding how dozens or hundreds of genes are inherited together and how that might impact the phenotype in a given patient. Now, what I've talked to you about so far are EQTL risk genes. So these are genes that are in very close proximity to a schizophrenia risk SNP. However, the DNA is not packed into our cells in a straight line. It loops and folds to package into the nucleus, and that will bring regions that are distal into closer proximity, linking faraway regions to schizophrenia risk variants. So to query this, a former MD PhD student in the lab, Prashant Rajarian, in collaboration with Shram Akbarian, did high-C genome-wide mapping across IPS-derived neurons, astrocytes, and neuroprogenitor cells. He then expanded the set of genes that we thought were linked to schizophrenia risk. So there are 224 genes in close proximity to schizophrenia risk genes, and a couple hundred more that are linked to risk loci by chromatin loops. And he was able to validate these by RNA-seq and by CRISPR. And so here, for example, at this one locus, we have 4 schizophrenia risk SNPs, 93 kilobase pairs away from the protocadherin alpha cluster. Prashant applied a Cas9-based strategy to delete these risk SNPs. And when he did, what he saw was significant changes in the disease-associated direction of 2 of these genes, protocadherin alpha A8 and 10. Now, moving in a slightly different direction, we'll talk about using IPS to explore the impact of rare deletions in schizophrenia. So here again is the most recent genetic analysis of deletions, copy number variant deletions in schizophrenia, and I'll talk to you about this one here at 2p16.3. Of all of these CNVs, this specific loci is unique for a couple of reasons. One, it's the only loci that includes just a single gene. 2, it's non-recurrent. The boundaries and the size of the deletion actually varies between patients. And 3, this gene is highly alternatively spliced with hundreds of predicted splice isoforms. So for example, this was the first mapping of Norexin-1 splice isoforms in mice. I'll show you this plot because despite our best efforts, we haven't found a better way to present the data either. The very top row is the most abundant Neurexin 1 isoform by long-read sequencing. The very bottom row is the least abundant. If an exon is included in a splice isoform, it's green, and if it's skipped, it's gray. So if you follow along the first exon, you can see include, skip. And basically you have long-read sequencing for every Neurexin 1 isoform. And in this paper, they found 247 splice isoforms in the mouse brain. Subsequent follow-up studies have been able to show that the number of isoforms is both brain region and cell type specific, with in total greater Neurexin 1 isoform complexity in regions of the brain with increased cellular complexity. So this was all done in mouse, and before we could begin to look at IPS-derived neurons from cases and controls, we had to understand what the Neurexin 1 isoform was in humans. And so this was work led by a former PhD student in my lab and a former postdoc in my collaborator, Gongfang's lab, Xuzha Zhu. So Aaron and Xuzha worked together first to undertake to compare the Neurexin 1 isoform repertoire in the human brain. Now we used a slightly more stringent isoform cutoff. We required that we saw an isoform independently 7 times to count it. So across 3 human brains, we report 66 high confidence Neurexin 1 isoforms, 83 high confidence isoforms in the fetal brain, and 68 high confidence isoforms in control IPS-derived neurons. And again, we show the data like this: the most abundant at the top, the least abundant at the bottom. And the important thing to note here is that most of the isoforms at the top are shared between the brain and IPS-derived neurons. They're colored in orange if they're shared. So I think in general, it says that the most abundant isoforms are shared, and thus IPS-derived neurons are perhaps a good model to explore the impact of patient-specific mutations on the isoform repertoire. So here's our cohort. It's small, but that's because these mutations are incredibly rare. So we have 2 patients, a mother and a son, with 5' deletions encompassing the promoter and first 2 exons, and 2 patients with 3' deletions. They're twin brothers, encompassing the second, third, and fourth from last exons. And I'll remind you, these are heterozygous deletions. They're non-recurrent. They differ between families. The first thing that Aaron did was a single-cell RNA-seq of IPS-derived neurons, and what she saw was most interesting. I'm not sure if you can see my cursor, but on the top left-hand corner, we have the glial cell clusters. Those brown ones in the middle are the immature neuron clusters, and these teal and lighter green cells are the mature neurons. They're circled in red. And what we see when we color them by donor is that these mature neuron cluster is disproportionately gray. In fact, we are disproportionately enriching for control neurons and depleting for patient-derived neurons in this mature neuron cluster, suggesting that patient-derived neurons might not be maturing to the same extent as controls. Aaron was able to show that there's reduced neurite branching from both the 5' and 3' cases, and that the neurons don't fire to the same rate by multi-electrode array. So as you can see, this is a time course of development, and the gray neurons start to fire more by week 4 or 5, and they really accelerate into weeks 5 and 6. And the patient neurons just don't fire at the same rate, and they don't seem to catch up. These are mixed forebrain cultures, but we could repeat the analysis using NGN2-induced pure glutamatergic neurons, and here you see it a bit more clearly. So again, control neurons ramp up activity over time, and the patient neurons just don't get to the same level. So the bigger question here is why? To what extent do these phenotypes reflect perturbations in few or many Neurexin 1 isoforms? Are patient-derived neurons firing less because one supercritical Neurexin 1 isoform is missing, or because all the isoforms are reduced by a smaller amount, or for some other reason altogether? So here's our Neurexin 1 profiling of case and control neurons. And what you'll see is that there's mass dysregulation. So half of the wild-type isoforms are significantly decreased in patient-derived neurons. This makes sense. They are heterozygous deletions. Another almost third of wild-type isoforms are not detected in our patient-derived neurons. And again, these are the less well expressed in controls. And so really what you're seeing is that many isoforms are decreased in patient-derived neurons, but you're also seeing this unexpected thing here. 31 unique mutant Neurexin 1 isoforms detected in the patient neurons, never detected in control neurons, and never detected in the human postmortem brain. So we cloned these mutant isoforms to understand if they had a functional impact and to see if they were translated. It turns out that overexpression of mutant isoforms in control neurons is sufficient to significantly reduce expression of control neurons. In those 5' patients, the ones with reduced orexin expression but no predicted mutant isoform expression, overexpression of a wild-type isoform was sufficient to rescue reduced neural activity. But in the 3' cases, we actually were unable to rescue activity. These were the patients expressing wild-type isoforms. And so overall, we think there's 2 things going on here. Neuronal phenotypes in Orexin-1 neurons occur through both a loss of Orexin-1 dose in all of the cases, and in a subset of cases, the additive effect of mutant isoform activity when they're formed. And we don't think this is an unusual effect unique to this one particular family. If you dive into the PGC data sets, it turns out about 1 in 5 patients with Orexin-1 deletions include coding regions in such a way that one might predict that mutant isoforms are formed. And so with the few minutes I have left, if that's okay, Pablo, come shut me up if it's not. I want to talk to you about drug screening, okay, and precision medicine. And so the goal here is to do smarter drug screening because it's so hard still to scale these experiments to hundreds or thousands of donors and hundreds of thousands of drugs. And so we worked with Joel Dudley to predict a smaller group of drugs that we thought might be sufficient to reverse schizophrenia signatures. And so he basically mined geo if you had samples from patients and controls. He did not care if they were skin samples, blood samples, or brain samples. He just used them to derive disease gene expression signatures. And similarly, if you had samples that had a drug treatment and a vehicle control, he didn't care if that sample came from primary cells or cancer cell lines. He just merged them to make drug gene expression signatures. And he integrated those to identify drugs that he predicted to oppose schizophrenia gene expression signatures. And that's where we started our drug screen. So we conducted this drug screen as a transcriptomic-based drug screen because we didn't believe we had satisfactory phenotypic markers to mark schizophrenia. So we did this in neural progenitor cells from 12 patients and 12 controls and 8 classical cancer cell screening lines across 134 predicted drugs that might reverse schizophrenia signatures. And this analysis was led by Ben Reedhead in collaboration with Eric Schad and Joel Dudley. And so here, what I'm showing you is that some of the time context matters. So there were some drugs like Selectatil, which is the top in every category. They had meaningfully and large effects in every cell type. But there were other drugs that had effects in cancer cell lines, but not in neural progenitor cells, or in neural progenitor cells and not in cancer cell lines. And so what we were trying to show or to understand is when is it worthwhile to look at patient-derived cells, and when is it worthwhile looking at the disease context. And the overall conclusion here was that you get value added. So there's a lot of information that is to be gained by screening in cancer cell lines, but the value added data that you get by looking at neural cells is enriched for schizophrenia risk variants and neuronal synaptic pathways. And so the real question here is, can we use this to help patients? Is drug responsiveness predictable? We don't necessarily have to find a drug that cures or treats all patients in order to do good. If your drug only helped a subset of patients, that would be okay too, so long as you could tell in advance which patients you should be giving this to. And so the question then, does genotype predict clinical response, or does in vitro drug response predict clinical response? Going back to that drug screen I showed you on the previous slide, we did have one group of patients with known drug response. There was a subset of the schizophrenia patients who had all been on a blinded clozapine trial. However, we did not see a meaningful association in clozapine-induced gene expression that corresponded to diagnosis. So we went back to the clinician who did this experiment. She was like, of course there's not. Everybody, Kristen, is less psychotic on clozapine. Some patients just don't handle the side effects as well, and that's why they failed the trial. So I think clozapine would be a terrible drug to try to predict for clinical utility. And so what would be a good drug? What's the gold standard drug where we think we might be able to predict drug response? Well, you would want that to be a drug where patient responsiveness is lifetime stable. And you would want it to be a drug whereby if a patient's father or uncle had the same disease and they responded to this drug, that the patient themselves are much more likely to respond. And that gold standard example is lithium responsive bipolar. So this was a study that I had the fortune to start as I wrapped up my postdoc, and the good fortune to hand off to Jerome Mertens, who did a really great job finishing. He was able to show that IPS-derived neurons from bipolar patients were hyperexcitable, but that hyperexcitability was reversed by lithium treatment only in lithium responsive patients. And then Rusty himself, in a second study, reproduced this finding in an independent cohort, and he was able to show that by looking at just 5 neurons from a patient, they could with very high confidence call whether that neurons came from a lithium responsive or non-responsive patient. And so with that, the big take-home message here, I hope, is that as we better genotype patients, as we better translate DNA and its epigenetic marker information to changes in gene expression and synaptic function, we might be able to inform treatment. I think that stem cells and CRISPR will be really useful in helping us understand this link between DNA and RNA, and then again from RNA to synaptic function, and that we could conduct drug screens, I hope you see, at both these points, at RNA or synaptic function, and that gives us really 2 points to predict clinical response. And so with that, this is my most important slide. This is everybody who connected the work and everybody who paid for it. Special thanks to Nadine, Aaron, Prashant, and Suk, who've all since left the lab, but largely drove all the stuff that I showed you today. And I'll note that we have open funded projects. So if you are looking for a postdoc at the moment, please reach out. Thank you.
So thank you so much, Kristen. It was an enlightening presentation. I really enjoyed it. We've got one question from YouTube, but I think that's a broad question. We can leave it for the last one. So I want to know if you guys have a first question to Kristen.
Yeah,
before.
I have some questions. First of all, amazing presentation. It's very exciting to see like how many different things you guys have been doing. And one thing that I was interested, it's maybe like 2 blocks of questions. The first one is, you mentioned in the beginning when you decided to stop competing with the geneticists and work with them, like, have you been considering using more complex models, like adding cell types, for example, microglia? And in the same line of this, like, have you considered looking at the effect of these specific mutations in microglia, for example? And I'm pointing microglia because of the C4 study that came out like about 2 years ago. And I think the second question that it's not super related to that is about the unique neurexin isoforms that I was also super curious about it. How do you think this is appearing? Like, would that be like an alternative splicing? Just would be interested in learning a little bit more about it. Thank you.
Can I just add a sub question to the second part of the neurexin? Because I was wondering what the mutations did to the protein itself, like molecularly, that to diminish, to reduce synaptic to spike number.
Okay, we're gonna see if I can remember all of these questions. Question number 1 was about adding a second cell type. So yes. In truth, I've been telling every student and postdoc in my lab the last couple of years that you must either add a second cell type or many more genes. The era of doing 1 gene and 1 cell type is over, and we are not doing it. It's taken some time to fully transition to this new way of going. And what I can tell you off of like an N of 10 now is that if you give people the choice between adding a second cell type or adding a second gene, they all pick extra genes. And so we are hugely pushing forward into some new methods like massively parallel reporter assays and CRISPR screening, because looking at not just how many genes we can perturb, but how many in combination, and the impact on not just RNA-seq, but MEA and synaptic imaging. And so a lot of our new efforts are in that direction, not because I necessarily think it's the only or the best, but because the people that I have hired have really want to push that way. We're doing some stuff in microglia for Alzheimer's, since a lot of the common variants there are actually really enriched. With schizophrenia, there's a signal, especially in complement, but not broadly beyond that. And so if you're going to do multiple genes, that will always drive you back to neurons. And so while we haven't done much microglia stuff in schizophrenia yet, we do have some cool stuff coming through for Alzheimer's. Norexin question. Yes, I thought it was alternative splicing. So we think that there is just a lot of splicing at this loci, and they are splicing around the deletions. And so you can see that every single mutant isoform in the patient was a splice around the deletion region. What is it doing to the protein? Well, the specific 3' cases we had, the deletion included splice site 4, I think, which is a canonical one and really important. And it has known, so Norexins are thought to be synaptic organizers. And I cut a bunch of this just in terms of time today, but they live on the one side of the synapse, but each Norexin 1 isoform binds to a different protein on the other side of the synapse. And so our thought is that if you are missing a region, and I think, for example, if the deletion encompasses the binding region for neuroligans, then you would change the composition of the synapse because specific postsynaptic partners would be missing. And it turns out that Norexin 1 isoforms don't predict where a neuron will migrate to and connect with. They predict what type of neuron it will be. And so it's possible that perturbing repertoires changes cell fate. And it's also possible that it changes the type of synapses being formed and the function later. And so a lot of what we're doing now is trying to dissect out whether the impacts on maturation and patterning or whether the impacts on synaptic stability and activity and function.
Listen, just complement a little bit what Pablo asked you. Very nice talk. When I saw you there in Munich, it was quite nice, the talk there, and I was adding to it. And I was wondering about Norexin, really, because, I mean, when you added the wild type one, you could rescue the phenotype. So do you believe that, and you were saying now that maybe it's lacking a piece and maybe it cannot connect anymore. But do you believe that that could also be like a gain of toxic function, like maybe it's acting as a dominant negative of the...
Yeah, so I think both are happening. So remember that most patients are not predicted to have Norexin 1 mutant isoforms because the deletions, this is a huge gene, it's like 100 kilobases. And if the deletion is in an intron and another intron and it falls out of frame, which is what 2/3 of the time you should screw up the frame. So 2/3 of the time you'll get a null protein and not a mutant protein. And 1/3 of the time statistically you should get a deletion from a non-coding to a non-coding that is still in frame. And so I think in many patients, it's a loss of function of the wild types. And I think that's why in those patients adding back, and keep in mind, we didn't pick a random wild type to add back. We topo-cloned the most abundant one out. And so if it was the most abundant one in that cell type, I guess it stands to reason it was the one that was the best one to add back. I don't know that if we had picked the lowest expressed Norexin 1 isoform, you would have got the same rescue. So it was a biased rescue, I guess, in that sense. And then the mutant ones, yeah, I think that they do act as a dominant negative, and I don't really understand how, but the fact that they could impair the controls really suggests that.
But have you tried to increase the expression of the mutant one? Just like to do some technique to increase expression?
We tried a lot by CRISPR-A and I, and the Norexin 1 locus is really, really resistant to perturbations in endogenous expression, which is why it's all been by lentivirus so far. And so when we overexpress the mutant isoform by lenti, it does perturb control neurons and makes them fire less.
Just one last question from YouTube. Peter is asking whether you can get both schizophrenia and autism. I think James can also jump into if he wants. And if you can get like a confused diagnosis between schizophrenia and autism. Yeah,
James, feel free to jump in. But the short answer is yes. Norexin 1 is the second most significant risk factor for rare CNV for schizophrenia and the most significant for autism. The childhood onset cohort that I showed you from Judy Rappaport, about 1/3 of the patients were diagnosed with autism prior to being diagnosed with schizophrenia. I think that if you are psychotic by definition, you cannot be autistic, but that I'll let Jim answer that. I don't know. I know that many of the clinical features can overlap, and Jim can go further than that.
Yeah, I don't think I can add much to it. I think this might be a situation where you need your genetics colleagues to really look into that. But yeah, I mean, there's been a long correlation, but I don't have anything more to add. With regards to the isoforms, though, with the long non-coding RNA BACHD1 that we looked at, there was a conserved exon 3 that was deleted, and when that got deleted, a brand new exon 4 got used. So I hadn't appreciated how common an event this might be. Your findings are amazing.
So thank you so much, Kristen and James, for your insights, and all speakers for their questions. So we are moving on to Steven's now. So Bruna, can you do the honors?
Yes. All right. It's a great pleasure and an honor to introduce Dr. Steven Hain. Dr. Hain got his bachelor degree in biological science in Federal University of Rio de Janeiro, and his PhD in biophysics also in the same institution. After that, Dr. Hain spent some time in San Diego, where he did his postdoc at the lab of Dr. Jared Chung, where he described genetic mosaicism in the human brain. After that, he returned to Brazil, and he soon became a full professor of Biomedical Science Institute at Federal University of Rio. Currently, he is also the Chief Research Officer at the DOR Research in Education in Rio de Janeiro. His research is focused on investigation of molecular and cellular metabolic mechanisms associated to psychiatric disorders and on the effect of psychedelics in the brain. In 2016, Dr. Hain and his group published one of the first associations between Zika virus infection and microcephaly using human pluripotent stem cells and brain organoids. I'd like to say that Dr. Hain is also an amazing scientific outreach. He does amazing scientific outreach, and I had the honor to be his student from the undergrad to my PhD. So Stevens, go ahead.
Thank you very much, Bruna, for the introduction. Thank you, Tiago and Pablo, for this great gathering. It's very nice to see all of these folks, like all of these colleagues, and I'm very honored also to be invited here to talk to you guys. So since like we are in the Zoom meeting for a while, I'm going to be short in my presentation, and hopefully we have some questions by the end. So I'm going to share with you my presentation. Okay. So what I'm going to do is to summarize some of our data regarding the effects, the biological effects of the Zika virus into the developing brain tissue. So Zika virus was discovered in 1947 in one forest called Zika in Uganda, Africa. And from that time, from 1947 to up to 2013, basically no big deal regarding people get infected with the Zika virus. It was not really being considered a very risk or harmful kind of virus. But since then, especially when it reached Brazil, there was a connection between the brain malformations and the Zika virus. While you have like lots of people get infected with the Zika virus, we are also seeing thousands of children, thousands of babies being born with this kind of malformations that could be characterized in this picture by the microcephaly. So colleagues from our institute, they were started to study and to receive the patients and also postmortem tissue from these babies. And they, doing some neuroimaging, they started to see what is highlighted in this green circle, that, for example, in this image of the brain, these white spots, they can be considered some kind of calcification. And thinking about a brain that's under development, these calcifications could be basically an interruption, an arrest in the neurogenesis. And because of that, we decided to use the same approach that was already presented by Kristen and by James and also by Fabiola, that's use of iPS cells. So basically you get these cells that get cells from the skin of some patients and also from the urine of others. And then we have a bank of those cells that can use to study several aspects of the biology of the Zika virus. So besides using these iPS cells that you can make them behave as astrocytes and neuroprogenitor cells and neurons, we also can grow those cells as these human brain organoids. This is just some examples of how we grow those cells, and you're also seeing a section of these brain organoids in the bottom right part of this slide. So with this kind of tools, we decided to check what would happen if we directly use the Zika virus to infect these brain organoids. On that time, it was not clear if like the microcephaly was associated with the Zika virus, or even if the effect was indirect. For example, the pregnant mothers, the women, they could be infected, and because of some kind of immune response, this immune response would be causing that kind of brain malformations. So we decided to infect directly these brain organoids and the also nerve stem cells with the Zika virus. So we found that like the neural cells, they can be infected with the Zika virus. So what we're seeing in the left part of the slide, in red, this is an antibody against the Zika virus, and in blue you're seeing the nuclei of these brain cells. And then the right part, you're seeing these brown dots. They are the virus particles inside these brain cells. When we follow the growth of these brain organoids that were infected with the Zika virus, we found that like after 10 days, there was a reduction in the growth of those brain organoids. That would be around, there was a reduction in 30 to 40%. So it was interesting because then we have a model to study this brain development that could be altered by the Zika virus. We published that paper, and then after that paper, or why we're doing that paper, we also decided to see what would be the proteins that could be altered by the infection. So for this, in this study, we combined the proteomics with other techniques to evaluate what would be the proteins that could going up and down after the infection. So we found that like the cells of the brain, they are hijacked for the viral replication, which causes chromosome instability, DNA damage, and then the arrest of the cell cycle, and also the interruption of the neurogenic program. So all of these proteins, they become very interested for us to try to understand what would be the consequences of this network interactive molecular relationship. That would be important to understand why the cells, they die, and what would happen with different cell types in the brain. So this paper was published. And then one thing that also we were interested in that time is that, okay, we know that like cells die in the brain after they're infected with the Zika virus, but is there any kind of difference among the cell types regarding the survival or the death of those cells? So we generate nerve stem cells, astrocytes, and neurons from these iPS cells. And then we found that we have, of course, the infection of nerve stem cells, that was the cells that were originally described as being infected with the Zika virus. But then we realized that like the astrocytes, they are much more infected than the nerve stem cells, and the neurons, they are barely infected in this model. And using this kind of approach, we basically tried to describe what would be the pathway, the cellular change that would be caused by the infection of the astrocytes with the Zika virus. So we showed that like there was an increase in the oxidative stress that could be reverted with ascorbic acid. We also show that, like, confirming that data from our other paper, that like there is DNA damage on those cells that could be followed by the increase in the 53BP1 and H2AX proteins. And also, I don't have time to show to you, we also showed that there was a huge change in the physiology of the mitochondria after the infection. So this cartoon basically summarized what we showed in this paper, that like the virus enters into the cell, replicates into the endoplasmic reticulum. Because of that, it's going to basically drain the mitochondria, try to generate ATP, and then they'll have a release in the oxidative stress that finally going to cause DNA damage and the death of the majority of these of these cells. We published this paper in the beginning of 2020, and basically describing all of this pathway, the intracellular pathway caused by the Zika infection in the human astrocytes. But one thing that was really in our mind since 2015 was that why you have the northeast of Brazil as the epicenter in the world for the cases of microcephaly associated with the Zika virus. Why the case of the Zika virus congenital malformations were basically presented in this poor area of Brazil, even if you compare with any other region in the world. So we raised the hypothesis that there will be some kind of a cofactor associated with the brain malformations in the northeast of Brazil that could explain this difference in the amount of malformations of babies being born with malformations from women that they were infected with the Zika virus. One thing that I realized that because of a friend of mine, that's Renato Molica, who was he's a professor at the Federal Rural in Pernambuco, is that Brazil, especially the Northeast, was suffering the worst drought in history since at least 1930, exactly between 2014 and 2016. When you have a drought like that, the amount and the quality of the water gets very poor. So basically, this is the kind of the water that's available for the poor people in the northeast of Brazil during the drought. Okay, and this drought you can see by this color that basically with a lot of algae and a lot of cyanobacteria. And so we decided to check what would be the kind of cofactor that could be eventually present in the reservoirs in which the poor people in Brazil, that's basically the people that were also having the babies with microcephaly, would have access. So we measure it. It was in collaboration with Flávio Lara from Fiocruz, Patrícia Garcês from the Federal University of Rio de Janeiro, and several other folks. Then we measure the amount the amount of cyanobacteria in the different reservoirs in the different regions of Brazil. So you are seeing here in the left graph that you have almost 50% of the reservoirs in the northeast of Brazil. They have more than 10,000 cells, 10,000 cyanobacteria cells per milliliter, comparing to the other areas of Brazil. And when you look specifically to one of the toxins that is produced by this cyanobacteria, that's this Haphidiopsis, you see that 50% of the reservoirs, they have this saxotoxin. And this saxotoxin, it's produced by these cyanobacteria as a response to the stress. So they are living in this very stressed environment that is basically this kind of water. So you have a phenomenon that's called eutrophization. So we increase the amount of the cyanobacteria, and they also increase the amount of saxotoxin. So we after confirming that there was a cofactor that was different, comparing the northeast of Brazil with other areas of the country during the epidemic of Zika, we went back to the lab and combined the Zika virus with saxotoxin in these brain organoids. What you're seeing here is this reconstruction of the brain organoid, and these green and blue spots, they are basically the areas of neurogenesis that can be followed within these brain organoids. So our question was, if we combine Zika virus with the saxotoxin, we would have an increase in the consequences of the Zika virus within the developing brain tissue? And the answer is yes. So the saxotoxin is able to increase Zika virus-derived cell death in the human brain organoids. So what you're seeing here in the image is in the blue, these are the brain nuclei. In the red, you're seeing the cell death. So basically you have more cell death when you have Zika virus combined with the saxotoxin. And one other thing that's interesting is that we also saw an increase in the copies of the Zika virus when the tissue was exposed to saxitoxin as well. Then we moved it to animals to try to replicate what we saw with the brain organoids. So this part of the work was developed in the lab of Patricia Garcés. So basically, what she did was that she feed the animals with these animals started to consume water with saxitoxin, basically the same kind of water that was available to the people in the northeast of Brazil, to the poor people in the northeast of Brazil. And after they were drinking this water, they got infected with the Zika virus and also got pregnant. And then after that, she started to evaluate brain malformations, comparing the pups from the pregnant mice that was receiving the water and the Zika with the others just with the Zika. And she found basically that you have more cell death, and they have also changes in the thickness and other malformations in the brain of those animals, comparing with the controls and with the Zika alone. This paper we published March this year, suggesting that like the cyanobacteria, saxitoxin, is able to increase cell death and also brain malformations induced by the Zika virus. If you guys are more interested in this line of research, there was a piece about our work in the Scientist in June of this year. So to summarize our data, basically we propose that like the cause of the amount of brain malformations that was the highest one in the world was caused by the lack of basic sanitation. So the lack of basic sanitation for the poor people in Brazil, especially during the drought, made these people to have to drink this water with saxitoxin, and the saxitoxin was able to make worse the malformations caused by the Zika virus. Of course, you see brain malformations in the other regions of Brazil and also in other areas of the world, but there's no comparison for what we saw in the northeast of Brazil with the other areas, even with the southeast of Brazil. Of course, this is not the only cause of these brain malformations, but we believe that this is one of the most important cofactors for the congenital syndrome caused by Zika in humans. This is, for example, another cofactor, that's malnutrition. This work was developed also by other colleagues in Brazil and Argentina and UK, showing that like protein malnutrition is also able to be associated with congenital Zika syndrome. This work was headed by Patricia as well. So with that, I finished my presentation. I'd like to be a little bit more short because of the time, because probably you guys are Zoom tired. So this is a picture of my team before, of course, the pandemics. So I really would like to thank all of them, especially in this paper, the last paper that I showed you, Carolina Pedrosa and Leticia Rocha, who they are the heading this project there in the lab. So after the pandemics, we are back on track. We're working with social distance, with all of this protection, testing and everything. So I'm just going to briefly mention to you, we have 2 preprints now submitted as well, of course, showing that like SARS-CoV-2 infection in the brain tissue of a 1-year-old child, and also trying to understand the kind of infection that that's happening in the brain using neurospheres. So I'd like also to thank the sponsors of my lab, and also thank my institutions, Federal University of Rio de Janeiro and the Door Institute. So with that, I finish my presentation. Thank you.
Thank you, Stevens. I think that was great. Maybe while the others start thinking about a question, I have a question for you. Did you guys tested the effect of this toxin also with the infection of dengue and other very common viruses that we have usually in Brazil?
No, we haven't tested the saxitoxin with any other kind of virus, but we believe that, like, if you have a part of the population being exposed to the saxitoxin, you make them more vulnerable to any other kind of disease. Just to give you a background, why we decided to look at the saxitoxin, in 1994, 1996, there was a huge number of deaths in exactly the same area, caused by with patients having hemodialysis, right? They were having hemodialysis, and the water that we used in the hemodialysis, it was contaminated with also with cyanobacteria. It was a different toxin. It was not saxitoxin. It was microcystin. But this is a very complicated thing that, like, happening in different parts of the world. Maybe also, of course, associated with the global warming, but that could affect different aspects of other diseases that are being associated depending on the region. Just as a curiosity, there was also a number of deaths in Africa of elephants because also one bloom of this saxitoxin, of this cyanobacteria. So it's something that we really have to keep in mind, that unfortunately the lack of basic sanitation and the availability of very poor quality water can make populations in the world more vulnerable to different diseases. In that case, we're seeing that, like, it was associated with Zika, but I think that would be interesting to see, as you suggested, to see, for example, the dengue, chikungunya, and even COVID.
Thank you so much, Stevens, for your talk. We've got one question from YouTube, but I'm gonna pop forward and I have my own. It's because we also have, like, really poor areas in Asia and Africa, and Zika virus is also kind of prevalent in Africa. And what do you think are the reasons for not having this much cases of brain malformations in Africa, for instance, or even in Asia, and only in Brazil?
It's a very interesting question, Pablo. One possibility is that, like, we have we had, at least, I don't know if you have anymore, but we had at least a good track system. I mean, we were able to follow the cases of microcephaly in a very good way. I mean, like, if there was a baby that was born with microcephaly, it was easy to track in Brazil. I don't know about the systems of tracking children, for example, combining Zika with malformations in the other parts of the world. The other thing is that, like, maybe we really had a perfect storm because of the droughts. So this drought was without comparison with any other period since 1930. So maybe this also could explain what happened in Brazil comparing with the other areas that you mentioned.
Okay. Thank you so much, Stevens. Sorry, go on.
May I ask one question, Pablo?
Yep.
Is that okay? Yeah. It's very nice, Stevens, that I've seen Patricia talking about the data about the poor nutrition, and it's very nice that you are looking to the poor water conditions that we have here in Brazil, and then we can bring awareness about this very important thing, right? That still takes place here in Brazil, unfortunately. And I was wondering if we're going to investigate a bit more, like, the mechanism behind this toxin, like, making things worse in terms of Zika. Maybe, do you think that's more related to the immune system? And, like, what do you believe? How do you think that this toxin makes things worse?
Yeah, that's the same question from Tarsila in the YouTube.
And kind of the same vibe. Do we have any, I don't want to say biomarkers, but maybe, like, some form of diagnosis if people do have this this toxin?
Okay, so I will start with Tiago, and then I move to the other question. It's very tricky to track the contamination with this kind of toxins. Some of them, they accumulate in the liver, and the others, they are very labile. So, for example, one thing that we submitted this paper to Science before it was published, and I'm very happy as it was published, but before we tried Science, and the reviewer asked, like, the editor asked, okay, so I'd like you to check if you have more contamination in the postmortem tissue of this babies with malformations. And we tried with several ways, but like it's so, all of the material that we had was fixed with paraformaldehyde. And it was impossible to track that in the tissue. So even in the tissue, or maybe like if you have to get the population, but since it was associated with the drought, it was very tricky to see, for example, after the drought, how long it will you have the kind of contamination in their blood, for example. So it's something that I would maybe it would be easy just to go and to see the check, for example, as we did, the quality of the water around the areas, because of that these people they only have that water to drink, and it's important to tell that like even if you boil the water, the toxin is still is destroyed for the water, but it's hard to follow in the body if you are going to be contaminated. So it's very tricky. So considering now the question of Pablo and Fabiola, yes, we indeed try to follow how the saxitoxin would be able to change the sensitivity of these brain cells to Zika virus. So we have two hypotheses. It's associated with the sodium channel. This is one thing that is being able to interfere with the sodium channel, that we know that sodium channel is important for the brain development, of course. And the other is this receptor that's called AHR, that's aryl hydrocarbon. That's one receptor, intracellular receptor, there was a nice paper in Nature Neuroscience from some folks from Brazil, including Jean-Pierre Perron from USP and others, showing that like this AHR receptor is critical for the development of Zika. And now we are trying to understand if AHR would be associated with that, and also the role of the sodium channel in this process. The other thing that I don't have at the moment how to share more of this data, it's still preliminary, but looks like that depending on the cell type, the effect is different. The consequences of the combination of the Zika virus with the saxitoxin is different comparing, for example, astrocytes with neurons and with neurostem cells. So, but all of them, they're bad consequences anyway, but they are different. So we're trying to understand if there is any kind of more in details the difference among those cell types, and also in terms of like the molecular speaking, what would be the consequence of this.
So thank you so much, Siemens, for your information, your presentation. I'd like to invite everyone back to the Zoom so we can like summarize and give a few words to people about the challenges in studying neuroscience in the future, because we are in a tough place right now that with the COVID-19 pandemic, and then for instance, signs that it interferes with the brain. So we'd like to hear about from you guys, what do you think that are the challenges to study neuroscience in the future?
That's an open-ended question.
Yeah, anyone can answer.
I mean, from my perspective, it's really easy to conceptualize how to connect variants to genes and genes to functions in a cell. But it's very hard for me to imagine how we're going to go from cells to circuits and behaviors. And so some of that might be organoids, some of that might be microfluidics, some of that might be 3D printing, some of that might be embedded arrays in like our fancy pants new, but the problem with all of these things right now is that I see the field diverging. There are people who are like getting very, very techy about making the best system possible, but those systems are not very manipulatable to look at as many genes as necessary. And so you already see it in the mouse models, right? You have mouse models based on genetics, like autism and schizophrenia. You have mouse models based on circuits, like addiction. And those hardly ever connect. And I fear that if we're not careful, we're going to separate our super cool, flashy, techy, organoid complex folks and the genetic folks who keep like regressing back to like the easiest cells to work with. And they made everybody sad. No one's sad.
Do you even get this PhD now?
Maybe just one thing that I would like to add is that, like, first to see how powerful and how interesting is neuroscience, and also for the students that they are like watching this gathering, how we can do neuroscience in different aspects and to understand a disease as schizophrenia, autism, and Huntington, and also maybe to try to understand a little bit more this kind of environmental changes that are associated with brain development. So I'm a little suspicious to talk about neuroscience because I really enjoy the field. I'm like on that field since the beginning of my career. And I think that also it's important for the students from Brazil to see that, like, especially as James was saying about their students there and the people from the country here, that we have a very good background and that, like, you can go to different places of the world. The science is international. And I think that this is some kind of relief if we think in the way that we're living in Brazil right now, because of the government, because of the challenge that we have, to have this kind of perspective that, like, we can go abroad. It's important. I think that, like, it's an interesting career, even if you're in Brazil right now, with the perspectives of the neuroscience, with the perspectives of the labs that we see here from James, from Chris, and from Fabiola. I think that it's a wonderful field to choose as a professional career.
Yeah, I can add to that as well. I mean, I think it's important to realize that you can move fields. Kristen said she wasn't trained as a neuroscientist. I wasn't trained as a neuroscientist either. I mean, my background was doing gene therapy for hemophilia, using viral vectors. And, I didn't really even get into blood stem cells at that point. And, you just follow what is an exciting question using the best systems that you can. And sometimes it's time to move on. It was around 2008 when I was realizing that the blood gene therapy work that I was doing was just losing its innovative edge, and I really couldn't move into the clinical side of gene therapy. So it was time to move on. And as it turned out, we had all the right expertise to move into iPS cells that were just breaking out at that point. And so, I think in probably all of our careers, talking of the scientists on the panel, we've all moved from one thing to another and taken advantage of what you learned in a different field, and you apply it to a new field, and maybe that new field hasn't thought that way before, and you bring something really new. So, your careers are not going to be just limited to what you're working on now. They're going to move in new directions. And the other aspect, I think, Kristen also, I think, commented on this, is that, no one system is going to be able to address all questions. And you have to really appreciate the value of people working on other systems. So the stem cell approaches that we take cannot tell us anything about behavior, and yet we're studying autism, right? And we're not studying behavior at all. And so what I found with previous stages in my career is that there are multiple ways of looking at different things. Sometimes the perspective that different scientists bring are really different, and they come with very different conclusions on the same topic. And what you really have to try and get to the core is, if my model explains my data, but it doesn't explain, Stephen's data, but Stephen's results are solidly done, then the model's wrong. And we've got to be thinking about new ways of thinking that actually integrate the data from multiple fields. And if the model that you're working on actually helps explain other people's data as well, not just yours, it's more likely to be right, and it's more likely to help your field move forward. So being very open to what other people are doing in other systems and the limitations of your own work is essential. That's what we need to be doing as scientists.
Well, thank you all. I think this symposium is really, and thank you so much, Pablo, for organizing and inviting these amazing speakers. This symposium is really finishing a pocket version of Clubes de Ciencia Brazil, which is one of the branches of a huge umbrella of Science Clubs International, which are programs in 9 different countries right now that really bring scientists from everywhere to inspire, especially high school students and beginning of college students to be scientists. And we had this amazing version this year virtually, of course, because of the pandemic. And it was very nice last night when we finished our activities. It was past midnight and we were with 200 students discussing science, and it was amazing to see how excited and hopeful they were, although most of them were super concerned about the situation of science right now, not just in our country, but also in the United States where I am, where science is not being well valued. And we were trying to tell them that this may be a phase and there's hope, science has to continue. So maybe before I end, if you guys had like a nice message that we could send to our students, I think they would appreciate a lot. And thank you again for being here, for taking this time to be here discussing amazing neuroscience. We had a club on neuroscience, which was super competitive, and we had amazing work done in the end by the students. So thank you again.
I would like to send a message to them. I think from this panel and like looking at all the techniques that we have to employ to study neuroscience, the thing that's more clear is that we need to collaborate and we need to work together like towards the goal of answering our questions. I believe that like working together is one of the main things in neuroscience for me, because you have a question, but you never have all the tools to answer that question. And so it's very important to collaborate. For instance, I talked to Kristen about a project I submitted. I work with Stevens to work with the iPSC cells, introducing the iPSCs in the lab. And I hope with James, we're going to start some collaboration. And I think that the main thing is that we have to cooperate and work together here in Brazil or abroad, and that will open opportunities for scientists.
Thank you, Fabiola.
I'm just going to say one phrase that I use with my students here all the time, is that like, let's be optimists. Let's leave the pessimism for better times. So I like to use this phrase here in Brazil right now.
So guys, sorry, go.
I think that these are hard years to be trainees. None of us train in a pandemic, and so none of us have actual empirical advice on how to help move forward in all of this. And I think the truth is that each of you has to navigate this in your own path. And so I would just encourage people to reach out to their mentors, their collaborators, their lab mates, and know that on average, we'll keep science moving forward. Maybe not in the way that we planned. Your PhD might not be the way that you planned. Your postdoc might not be the way that you planned. But if you can take the skills that you had before and work with the people around you and be forgiving of yourself and your teammates, I would just encourage you to hang on and be okay.
Yeah, that's a great message. I mean, all of us who run labs, as Kristen said, we don't have this experience either. Well, we did have SARS in Toronto in, what was it, 2013, and that did shut us down. So, you know, maybe we did have a little experience then, because it was a really bad situation in Toronto. But that did not have any second wave, and it didn't really spread globally. But yes, the PIs and labs really feel for everybody, and we have families. We have personal experience with this as well. My son just finished undergrad and is not really too sure what to do. You know, contact tracing seems to be an opportunity that just came up in Toronto, and he applied for that yesterday. So, you know, maybe there are some short-term opportunities that you wouldn't normally consider. But at least in my group and my institute, the one priority is to preserve people's positions as long as we can, and funding is probably going to be a little bit harder. Some people are pivoting to COVID. Stevens, has a great opportunity for that. But other people are just looking to maintain their training and help out and keep everyone safe. Like Kristen says, there's all sorts of support mechanisms that are activated in the universities. And if you're struggling, even just from, personal isolation, please reach out and get every resource that you can. And hopefully, science moves incredibly quickly with the COVID. I'm a little skeptical that vaccines are the solution because, no vaccine has actually worked in a coronavirus previously. And, it's sort of percentages that work in flu, but I'm not an expert in this. But, at the same time, treatments do seem to be moving forward quickly, and doctors know how to treat things better. And I'm optimistic that by, next Christmas, we'll be through the worst of this and lives can maybe normalize. And I hope yours does.
Thank you all for attending this symposium. We are really, really glad that you accepted our invitation, even though situation with the pandemics makes everything hard for everyone. And that you took a little bit of our time to spread the word about science. I mean, we are in the difficult position right now, both with the pandemics and the denial of the science as a tool solution. So thank you all for joining us. Thank you all for watching on YouTube. The video will stay recorded in our platform. I'm gonna send you guys the link if you want and to watch later, and see you guys soon. And hopefully everyone, the next time we see each other, we are together in a physical place. Thank you guys.
Thank you, everyone. Thank you for organizing it. It's been a great meeting.
Yeah, you guys did a great job.